From the very first moment of waking, before the eyes have fully adjusted to the light, before the first word has been spoken or the first decision made, a quiet chemistry is already in motion deep inside the brain. It begins in the dark interior of a single neuron, in the ordinary and relentless work of staying alive. A nerve cell consumes energy the way any working thing consumes energy, steadily, without pause, drawing on a molecule called adenosine triphosphate, burning it down to power the electrical signals that make thought and motion and memory possible. And as it burns, it leaves something behind. A smaller molecule, stripped of two of its phosphate groups, drifts out through the cell membrane into the narrow fluid-filled space between cells. It is a byproduct, a remnant, the chemical equivalent of ash left after a fire. Its name is adenosine. One molecule means nothing. One neuron releasing adenosine into the fluid surrounding it is an event too small to feel, too small to measure except with instruments designed for exactly that purpose. But there are roughly 86 billion neurons in the brain, and they are all working, all burning fuel, all releasing this same small byproduct into the same extracellular fluid. And so from the first minute of every waking day, a slow tide begins to rise. It rises in the basal forebrain first. The basal forebrain sits deep at the base of the brain, just above where the two hemispheres meet the brainstem, and it is one of the brain's primary stations for managing wakefulness. Its cells are sensitive to adenosine in a precise and measurable way. They carry on their outer surfaces receptor proteins shaped to receive this molecule, two types, called A1 and A2A, and when adenosine binds to them, those cells begin to quiet, their firing rate drops, their influence on the broader arousal network weakens in small increments, hour by hour, in direct proportion to how much adenosine has accumulated in the fluid around them. This is not a metaphor. The concentration of adenosine in the extracellular space of the basal forebrain is a real quantity, measurable in micromolar fractions, and it rises in a real and predictable curve across the waking day. The brain is, in this precise chemical sense, keeping count of its own wakefulness. Every hour of consciousness adds another increment to the total, and the total accumulates toward a threshold, and at the threshold the pressure toward sleep becomes difficult to resist. The molecule that does this is not exotic. It is not produced by some specialized gland at some particular hour. It is produced everywhere, always, as a simple consequence of the brain doing what brains do. And in this sense, the need for sleep is not a weakness in the system. It is the system functioning exactly as it was built to function, accumulating the evidence of its own work, and eventually insisting on rest. There is a word for this accumulation, for the rising pressure toward sleep that grows with each waking hour. It is called homeostatic sleep pressure, or process S, the S standing for sleep. A Swiss scientist named Alexander Borbély, working in the early 1980s, was the first to describe it with the mathematical precision the phenomenon deserved. He was interested in a question that seems simple on its surface, but had never been cleanly answered. What actually determines when a person falls asleep and when they wake up? Not the social pressures, not the alarm clock, not the cultural habit of sleeping at night, but the underlying biological forces that govern the timing of sleep in a body left to its own rhythms. What Borbély found, working through carefully controlled studies of human sleep patterns and EEG recordings, was that two separate processes were operating simultaneously, and that their interaction explained almost everything about when sleep arrived and when it ended. The first process was the one already described here, the slow, steady, exponential rise of homeostatic sleep pressure across the waking day driven by adenosine and reflected in the mounting pull toward rest that anyone can feel by early afternoon. The second process was something different in kind, something that operated not on the hours since waking, but on a cycle all its own, independent of whether the person was asleep or awake at all. Borbelli called this second process the circadian alerting signal, or process C, and to understand it, the attention must move from the basal forebrain to a structure positioned just slightly forward and above it in a paired cluster of roughly 20,000 cells that together form one of the most precisely regulated clocks in biology. This structure sits directly above the point where the two optic nerves from the eyes cross each other on their way into the brain. The crossing is called the optic chiasm, and the two small nuclei positioned just above it are called the suprachiasmatic nuclei. Each is smaller than a grain of rice. Together, they are the brain's master clock, the central timekeeper, around which all the body's other biological rhythms are organized. Their cells fire with electrical rhythms that cycle with a period of approximately 24 hours, rhythms that were already cycling in the womb before the eyes had ever seen light, and that have been running continuously ever since in every person alive. These cells pulse. They are not simply on or off, they oscillate, their electrical activity rising and falling in a smooth wave that repeats day after day with extraordinary regularity. And the rising part of that wave, the part that corresponds to the waking day, generates what Borbelli identified as the circadian alerting signal, a continuous broadcast of electrical influence that spreads from the suprachiasmatic nucleus outward to the arousal systems of the brainstem and hypothalamus, actively counteracting the rising pressure of adenosine, holding wakefulness in place against the growing chemical tide. During the morning and early afternoon, this alerting signal is strong. It is strong enough to keep a person alert, even as adenosine climbs, even as the homeostatic pressure builds, because the circadian system is actively pushing back. But the circadian signal is not flat across the day. It has a shape, a rhythm of its own, and that rhythm includes a predictable dip in the early afternoon. Between roughly 1 in the afternoon and 3 in the afternoon, the circadian alerting signal weakens, not drastically, not permanently, but measurably and reliably. The wave drops into a trough, and when it does, homeostatic sleep pressure and circadian drive stop balancing each other and briefly tip into imbalance, a window, a few hours wide, when the biological forces that keep a person awake are at their daily minimum, and the forces pressing toward sleep temporarily gain the upper hand. This afternoon trough is real, and it is not caused by lunch. That distinction matters because the most common folk explanation for afternoon drowsiness is the diversion of blood to the digestive system after eating, an explanation that is both intuitive and wrong. Studies conducted in conditions of complete fasting show the same dip at the same time, driven by the same circadian oscillation, with no meal in sight. The trough is in the architecture of the clock itself, not in the stomach. It would arrive whether the person ate or not. In societies that have built their culture around sleeping in the early afternoon, the siesta cultures of the Mediterranean and Latin America, the riposo habits of hot climates, this biology was recognized long before anyone had the tools to measure it. The body already knew. The afternoon dip shaped behavior across centuries. The neuroscience simply measured what behavior had long since implied, confirming that the clock built into those 20,000 small cells had been generating the same signal in the same window every day, long before anyone was watching. Now consider what this means for the average waking day. A person wakes in the morning with adenosine concentrations that have largely cleared during the night, with the circadian alerting signal beginning its morning rise. The system is in relative balance, and alertness is high. Through the morning, adenosine climbs and the circadian signal climbs with it, and the two processes remain more or less in step, with the circadian signal dominant and wakefulness well supported. Then, in the early afternoon, the circadian signal dips while adenosine, having been accumulating for 6 or 7 or 8 hours, remains elevated. The gap between what is pressing toward sleep and what is holding it back narrows to its smallest point of the day, and the pull toward rest deepens. Later in the afternoon, the circadian signal recovers. It rises again toward its early evening peak, temporarily pushing back against the adenosine load, generating what many people experience as a second wind of alertness in the late afternoon. Then, as evening deepens into night, the circadian signal begins its long fall toward the trough of the nocturnal nadir, and adenosine, having accumulated through a full waking day, reaches its maximum concentration, and together they tip the system irresistibly toward sleep. This is Borbély's two-process model, process S and process C, the homeostatic tide and the circadian wave, operating together, their interaction tracing out the full arc of a waking day, from the first clear-eyed alertness of morning, through the drift of afternoon, and on toward the deep pull of late evening. The model was formalized in 1982, and in the decades since, the evidence behind it has only deepened. The specific molecules, the specific receptor types, the precise anatomy of the pathways involved, all of it has been mapped with increasing resolution, and the map fits the model with extraordinary fidelity. But understanding when sleep pressure rises and when the circadian signal weakens is only part of the picture. The other part is understanding what governs the actual transition from waking to sleep, the mechanism that makes the change so fast, so complete, and so difficult to arrest once it begins. That mechanism is built into the hypothalamus in a circuit of remarkable elegance and simplicity. The hypothalamus sits at the very base of the forebrain, a small region dense with nuclei governing the body's fundamental regulatory systems: temperature, hunger, thirst, and sleep. Within it, there is a cluster of neurons in a region called the ventrolateral preoptic nucleus, whose activity rises as adenosine accumulates and as sleep pressure builds, and whose influence on the rest of the brain is profoundly inhibitory. The ventrolateral preoptic nucleus sends inhibitory signals to the arousal centers of the brainstem and hypothalamus, to the clusters of cells that produce norepinephrine and serotonin and histamine, the neurotransmitters of active wakefulness. And those arousal centers, in return, send inhibitory signals back to the ventrolateral preoptic nucleus. Each side tries to silence the other. Each side pushes against the other constantly. The result is a circuit that engineers call a flip-flop switch. The flip-flop is a switch with no stable middle position. When one side is stronger, it suppresses the other enough to release its own inhibition, which makes it stronger still, which suppresses the other side further, which tips the system decisively and rapidly from one state to the complete opposite state, wakefulness to sleep, or sleep to wakefulness, with very little time spent in between. The transition is fast because the architecture is designed to make it fast. A prolonged middle state, a brain half asleep and half awake, uncertain which way to go, would be dangerous. The flip-flop switch prevents it, keeping consciousness either fully on or fully off with clean boundaries between them. The light switch is not a bad analogy, though it is a mechanical one. A light switch stays off or on. It does not rest in the middle. The hypothalamic flip-flop circuit achieves the same stability through mutual inhibition rather than through a mechanical catch, but the effect is the same: a stable on state, a stable off state, and a brief decisive transition between them. What adenosine does in this framework is shift the balance of that switch across the waking day. As adenosine rises, it increasingly activates the ventrolateral preoptic nucleus, tipping the balance gradually toward the side that favors sleep, until the switch crosses its threshold and snaps over. There is one more element in this circuit that deserves its own attention because it adds a layer of stability beyond what the flip-flop alone provides, and because its role in the afternoon change in alertness is more direct and more specific than any of the mechanisms described so far. Deep in the lateral hypothalamus, in a region immediately adjacent to the areas governing hunger and metabolism, there is a population of neurons that produces a signaling molecule with two closely related forms. The neurons themselves number roughly 70,000 in the human brain, a small population by any standard, a fraction of the brain's total cellular count, but their projection fibers reach virtually every arousal center in the brain. They extend to the locus coeruleus, where norepinephrine is produced. They extend to the dorsal raphe, the source of serotonin. They extend to the tuberomammillary nucleus, which produces histamine. They extend even to the basal forebrain, cholinergic neurons, and to the cortex directly. No single arousal center sends projections as broadly as these cells do. They form a kind of distributed stabilizing current, running simultaneously through all the major circuits that keep a person awake. The signaling molecule these cells produce is called orexin, sometimes also called hypocretin in the clinical literature, though the names refer to the same molecule. Its primary function is not to create wakefulness from nothing, but to stabilize it, to hold the flip-flop switch firmly on the waking side, to prevent spontaneous transitions into sleep at inappropriate times, and to maintain the coherence of the arousal state against the constant chemical and physiological pressures that would otherwise pull the system towards sleep. The orexin neurons fire in tight, fast bursts during the morning hours. Their activity is detectable as rapid electrical volleys spreading outward through those broad projection fibers, a stabilizing current flowing continuously through all the arousal centers simultaneously, like a distributed signal holding a complex network in a single coherent state. When they fire, the locus cerebellosus fires. When they fire, the dorsal raphe nucleus fires. The brain's arousal systems are not simply running independently, they are being held in synchrony by this small population of hypothalamic cells projecting influence in every direction at once. But orexin neurons are not immune to the chemistry of the day. Their firing rate is sensitive to the same factors that govern the overall sleep-wake balance, to adenosine, to the circadian signal, to metabolic state, to light. And as the afternoon progresses, as the circadian alerting signal from the suprachiasmatic nucleus dips into its early afternoon trough, the orexin neurons begin to quiet. Their firing rate falls. The stabilizing current running through all those arousal centers weakens. The locus coeruleus, no longer being driven as firmly, reduces its output of norepinephrine. The dorsal raphe nucleus, no longer being held in its alert state firing pattern, relaxes. The arousal network, taken as a whole, loses some of its coherence, some of its mutual reinforcement. This is not catastrophic. A healthy adult does not simply fall asleep at 2 in the afternoon involuntarily, though some people come close. What happens instead is more subtle. The threshold for sleep drops. The flip-flop switch normally held firmly on the waking side by the combined influence of the orexin system and the circadian alerting signal, becomes easier to tip. The margin between the waking state and the sleep state narrows. Minor factors that would not matter at 10 in the morning, a warm room, a heavy meal, a soft chair, a dull task, a few minutes of quiet, become enough to tip the balance, because the buffer that normally holds wakefulness in place has become thinner. It is worth pausing here to let the geometry of this feel clear. The waking brain at its morning peak is a system held firmly in place by multiple interlocking mechanisms, all running at their strongest simultaneously, the adenosine load still low, the circadian alerting signal rising, the orexin system firing vigorously, all the arousal centers being held in active synchrony. By early afternoon, each of those mechanisms has shifted. Adenosine has been rising for 6, 7, 8 hours. The circadian signal has entered its daily dip. The orexin system has quieted, in step with the circadian dip. The arousal network has lost some of its morning coherence. The system is still running, still maintaining wakefulness, but it is doing so with less margin, less redundancy, less resistance to disturbance. A single neuron releasing adenosine into the fluid surrounding it is an event too small to feel, but 8 hours of that process, across 86 billion neurons, adds up to something measurable in micromolar concentrations, and measurable in the growing weight behind a person's eyes, and measurable in the slightly reduced sharpness of their thinking, and measurable in the fact that a horizontal surface and a few minutes of quiet feel more appealing than they did at 9 in the morning. The afternoon trough is in this sense the convergence of several slow-moving processes, all arriving at the same point of the day simultaneously. The adenosine tide has been rising since the moment of waking. The circadian wave has dipped into its early afternoon valley. The orexin system has quieted in step with the circadian signal, and the net result is a window, reproducible, predictable, measurable in individual brains, when the biological conditions for sleep are closer to being met than at any other point in the waking day. The suprachiasmatic nucleus, those 20,000 cells pulsing with their 24-hour rhythm above the optic chiasm, has been generating this same dip at the same point in the daily cycle for every day of a person's life. The rhythm was present before birth, established in the developing brain before the eyes had any light to report, before the concept of daytime or nighttime had any meaning. After birth, light through the retina refined the clock's timing, anchoring it to the local day-night cycle and keeping it precisely calibrated. But the clock's internal rhythm, its built-in tendency to generate a trough in the early afternoon, predates all of that. It is not learned. It is not cultural. It is in the architecture of the cells themselves, in the molecular feedback loops that these cells run continuously, loops that have been cycling without pause for decades. The loop is molecular at its base. Inside each cell of the suprachiasmatic nucleus, a set of proteins drives a cycle of gene expression and protein synthesis and protein degradation that takes approximately 24 hours to complete. Proteins produced during one phase of the cycle accumulate until they suppress their own production, then degrade, then allow production to begin again. This loop has no external input in the simplest case. It runs on its own. Light through the eyes can shift its phase, can calibrate it to the environmental day, but the loop itself runs whether light reaches it or not. That is why the afternoon trough persists in total darkness. That is why it persists during fasting. That is why it persists when the calendar and the social schedule say nothing about when rest should come. The 20,000 cells in the suprachiasmatic nucleus do not consult the schedule. They run their loop, they generate their signal, and the rest of the brain responds to it the way everything downstream of a clock responds to the clock, according to timing it did not choose and cannot override by decision alone. There is something grounding in this, something that shifts the moral weight of afternoon drowsiness. The person who wants to close their eyes at 1:30 in the afternoon is not failing some standard of alertness or productivity. They are responding to a chemical state in their basal forebrain, to a circadian signal generated in cells that have been running the same cycle since before they were conscious of anything, and to a quieting in a population of 70,000 hypothalamic neurons that are doing exactly what they are supposed to do at exactly the hour they are supposed to do it. The urge to rest in the early afternoon is not a personal weakness. It is a precise, measurable, elegantly coordinated biological event unfolding in the brain exactly as it was built to unfold. Caffeine, the most widely consumed psychoactive substance on Earth, works by sitting in the adenosine receptor without activating it, blocking the site so that adenosine cannot bind, preventing the signal of accumulated wakefulness from getting through. It does not reduce adenosine. It does not accelerate its clearance. It simply holds the door closed so that the message cannot be delivered. When the caffeine clears, adenosine rushes back in, sometimes with a rebound effect that makes the crash feel worse than the fatigue it delayed. The coffee is not addressing the chemistry. It is postponing the reckoning with it. The adenosine tide was real before the first cup and is still real after the last one. The adenosine, meanwhile, does not care about the caffeine. It continues accumulating, continues rising toward the threshold that will eventually bring sleep, continues binding wherever it finds an open receptor. By early afternoon, its concentration in the basal forebrain has been rising for 6, 7, or 8 hours. The number seems small in micromolar fractions, but the effect at that scale, operating on hundreds of millions of receptor proteins distributed across the basal forebrain and beyond, is not small at all. It is the entire physiological weight of the morning pressing on the afternoon. It is the chemical record of every thought and decision and perception since waking, written in a molecular residue that accumulates with every passing hour. The brain that sits inside a person's skull at 2 in the afternoon carries all of that history in its chemistry. It has been working since the moment of waking, burning energy, releasing adenosine, building pressure, while the circadian clock above the optic chiasm has been generating its daily arc, rising through the morning, dipping in the afternoon, rising again toward evening. And the orexin neurons in the lateral hypothalamus have been holding the arousal network together through the morning and loosening their hold as the afternoon deepens. Their small population of 70,000 cells doing quietly and continuously what they have done every day since early development, stabilizing wakefulness when it needs stabilizing and releasing their grip when the biology of the day asks them to. No single one of these processes produces the afternoon dip alone. Adenosine alone would produce a monotonic decline in alertness from morning to night without the midday second wind or the early morning peak. The circadian signal alone would produce a rhythm that had nothing to do with how long the person had been awake. The orexin system alone would provide stability, but no timing. It is the interaction of all three, the chemical tide of adenosine, the circadian oscillation from the suprachiasmatic nucleus, and the moment-to-moment stabilization from the orexin neurons, that produces the specific shape of the waking day. The morning clarity, the early afternoon dip, the late afternoon recovery, the deep pull of night. Borbély's two-process model captured this interaction in mathematical form in 1982. The experimental tools available to him at the time were limited compared to what came later: EEG recordings, plasma measurements, polysomnography, but the model has held up with remarkable fidelity across 4 decades of increasingly detailed molecular and circuit-level investigation. The things that have been discovered since have added mechanism and resolution to the picture, not changed its fundamental shape. The two processes are real, their interaction is real, the afternoon trough is real, and the adenosine that drives process S is as real as any molecule in the body, as measurable as blood pressure, as responsive to the hours of waking as a gauge responding to the pressure behind it. Consider for a moment the basal forebrain at the far end of an 8-hour waking day. The adenosine concentration there has been rising since the first neuron fired that morning. Its A1 and A2A receptors are heavily occupied. The cells that project wakefulness-promoting signals to the cortex are quieted. The ventrolateral preoptic nucleus, sensitive to the adenosine signal, has been growing more active across the day, pressing more firmly on the arousal centers, beginning to tip the flip-flop switch in earnest. The circadian alerting signal has passed through its afternoon trough and begun its secondary rise, but adenosine is rising faster now, and the evening is coming, and the circadian signal will soon begin its own long fall toward the nadir of the night. All of this is happening simultaneously, in real time, in the brain of someone sitting quietly anywhere in the world, reading or thinking, or simply resting, feeling the familiar pull of late afternoon without necessarily having the words for what is pulling. And through all of it, in the lateral hypothalamus, the orexin neurons are quieting. They have been firing through the morning, their projections carrying the stabilizing signal outward to every arousal center, holding the flip-flop on the waking side. Through the early afternoon, they have begun to ease. Now, as the afternoon deepens and the adenosine pressure builds, their firing rate falls toward its daily minimum. The stabilizing current weakens, the arousal network loosens, the locus coeruleus slows, the dorsal raphe relaxes. The histamine cells in the tuberomammillary nucleus reduce their output. All the components of the arousal system release their morning tension, step by step, as those 70,000 small cells in the lateral hypothalamus gradually, quietly fall still. The flip-flop switch stands poised. The chemical tide is high. The clock has turned toward evening. The stabilizing current has faded to almost nothing, and the margin between waking and sleep has narrowed to its thinnest point since morning. Everything in the biology of the afternoon has been moving toward this moment, in the same way that every slow tide has been moving toward the same shore, drawn not by any single force, but by the accumulated influence of many forces, all arriving in the same direction at the same time. The brain that reaches this point is not broken, it is not weak, it is precisely, beautifully, chemically prepared for rest, in exactly the way it was built to be prepared, at exactly the hour the ancient architecture of its rhythms intended. The orcs and neurons have done their work, held the day together through the long hours of the morning, and now they rest, and the day that they held in place rests with them, gently and without resistance, into the quiet that the chemistry has been building toward since the very first light of the morning. At the very edge of sleep, in the minutes before the world goes quiet, the brain changes the way it talks to itself. The shift is not felt as loss. It arrives more like a slow tide withdrawing from a beach, pulling small details back with it. The specific texture of the surface beneath the body, the particular angle of light through a window, The precise pitch of whatever sound lingers at the edge of the room. These things are still there. They have simply moved further away. And in the electrical record of the brain, during those first retreating seconds, the change is visible as a distinct alteration in pattern, a shift from one kind of wave to another, as legible as a change in weather seen from above. During ordinary wakefulness, the brain generates a low amplitude, high frequency signal across the scalp. Neurons across different regions fire in rapid, largely independent patterns, each area pursuing its own computation, the collective result reading on an EEG trace as a fine, busy, desynchronized scrawl. This is the electrical portrait of a brain doing many things at once, its activity spread and complex and awake. Then, as the first stage of sleep begins, that pattern begins to soften. The fast, irregular signal slows. The amplitude rises just slightly. A particular kind of oscillation begins to appear, emerging first in bursts, and then more continuously in the frequency range that falls between 4 and 8 cycles per second. These are theta waves, and their arrival on the EEG trace marks the transition from wakefulness into what is called stage 1 sleep. The eyes close, the muscles release a little of their held tension, external things recede, and the brain, which has been conducting its busy, distributed conversation for hours, begins to settle into something different. Stage 1 is shallow and brief, lasting only a few minutes in most people. It is less a sleep state than a corridor between waking and the first substantial sleep, a liminal space in which awareness of the external world decreases, but the body has not yet committed fully to being asleep. A sound, a shift in light, the sudden awareness of one's own falling, can pull the sleeper back without effort. The brain's electrical pattern during stage 1 is not dramatically different from drowsy waking. The theta rhythm is there, mixed still with the remnants of waking alpha activity, the characteristic 8 to 12 Hz rhythm that appears with closed eyes and a relaxed but still conscious mind. The transition is not a wall but a gradient, and crossing it leaves little trace. Stage 2 is something else. The arrival into stage 2 NREM sleep is marked on the EEG by two events that are among the most recognizable patterns in all of sleep science. The first is the K-complex, a single, large biphasic wave that rises sharply from the background and then falls again in less than a second, tall and unmistakable against the lower amplitude activities surrounding it. The second is the sleep spindle, a brief burst of rapid, rhythmically waxing and waning, oscillation appearing almost immediately after the K-complex, with a frequency between 12 and 15 cycles per second, building in amplitude over its first half second, and then diminishing over the next, the whole event lasting somewhere between half a second and 3 seconds before it disappears back into the flatter background. Together, these two signatures, the sharp declarative shape of the K-complex and the spindle's brief accelerating and decelerating rhythm, announce that the brain has entered a qualitatively different territory. Something organized is happening now, something that could not happen while the brain was awake. The K-complex is generated across the cortex as a coordinated event, a brief synchronous downstate in which many neurons fall silent at once, followed immediately by a rebound upstate in which they fire together. What triggers any individual K-complex is not always clear. Some appear to arise in response to external sounds or physical sensations registered at the threshold of awareness, the sleeping brain briefly addressing something it has detected without bringing it all the way to consciousness. Others arise spontaneously, as though the sleeping brain periodically checks the interior, issues a brief signal, and receives the silence of a stable environment in return. What matters more than the trigger is the form of the response. A sleeping brain that generates K complexes is a brain actively protecting its own sleep, consolidating its hold on stage 2 in the face of a world that has not stopped producing events. The spindle that follows is generated not in the cortex alone, but in a loop between the cortex and the thalamus, the brain's central relay structure. The thalamus sits deep within the brain, wrapped on its outer surface by a thin curved shell of inhibitory neurons that constitute the thalamic reticular nucleus. During waking, this structure acts partly as a filter, regulating what sensory information passes onward to the cortex. During stage 2 sleep, the reticular nucleus takes on a different role. Its neurons begin firing in rhythmic, synchronized bursts, and these bursts propagate inward through the thalamocortical relay neurons, which carry the rhythm up to the cortex in rippling waves. The cortex receives these waves and responds, its activity briefly rising and then subsiding in synchrony with the thalamic oscillation. The result, visible at the scalp, is the waxing and waning spindle, a rhythm built between two structures in a loop, each driving the other, the whole event suspended between them like a briefly sustained chord. During a nap of 10 to 20 minutes, a sleeper may accumulate between 20 and 40 separate spindle events, each one a brief instance of this thalamocortical synchronization. The spindles appear scattered across the EEG trace, like small distinct bursts of organized activity, each with its characteristic shape, each representing a moment in which the sleeping brain is actively doing something that has no equivalent in waking. These are not gaps in brain activity. They are not pauses in consciousness. They are a form of neural work that happens only in this state, in this particular darkness of stage 2, accessible to the brain only after the waking world has been let go. A nap that ends within 20 minutes ends here in stage 2. The sleeper wakes from a shallow, organized, spindle-rich sleep into a state of relative clarity, without significant grogginess, because the brain has not descended further than this first coherent sleep stage, and the transition back to waking requires crossing only a small distance. This is the range within which the so-called power nap operates, brief enough to preserve this shallowness, long enough to accumulate meaningful spindle activity. The brain's stare in stage 2, ending before the deeper pull of the next stage becomes decisive. What stage 2 accomplishes in this short window is real and measurable, and it belongs specifically to this state. To enter the next stage is to enter a different country altogether. Somewhere around the 25 to 30 minute mark, for a napper with a moderate accumulated sleep pressure, the architecture begins to change. The spindles become less frequent. The background EEG activity, which had maintained a relatively low amplitude during stage 2, begins to shift. Slow, large waves start appearing, delta waves, oscillating at between half a cycle and 4 cycles per second, of enormous amplitude compared to the fine-grained waking signal. These waves, wide and deep, represent something fundamentally different from the spindle activity of stage 2. During a delta wave, large populations of neurons across the cortex synchronize into a brief down state, firing together, then falling into a collective silence, then rising to fire again. Millions of neurons participating simultaneously in this slow oscillation, their coordinated activity sweeping across the cortex in a rhythm that is, by every ordinary measure, the opposite of waking thought. When delta waves occupy at least a fifth of the EEG signal within a standard scoring interval, the brain has entered stage 3, NREM sleep, the state also called slow wave sleep, the deepest the brain reliably reaches in a normal night, and the stage that a longer nap will enter if the conditions allow. The body during slow wave sleep is profoundly disengaged from the outside world. Sensory thresholds rise sharply. Heart rate and breathing slow and regularize. The muscles lose tone. Blood pressure falls. Growth hormone is released in pulses from the pituitary gland. The brain's metabolic rate drops. And in the EEG record, those wide, slow delta waves continue, each one rising and falling over the span of 1 to 2 full seconds, and each one representing a moment of synchronization across hundreds of millions of neurons, and then the silence that follows, a silence that briefly resembles the stillness of no thought at all. This is the depth that carries the greatest risk for a napper. Waking from stage 3. Slow wave sleep is a different kind of emergence than waking from stage 2. The brain, in slow wave sleep, is deeply engaged in the coordinated slow oscillation, and pulling it out of that state requires something closer to a forceful interruption than a gentle surfacing. The result, when it happens, is a period of confusion, slowed thought, and impaired function that can persist for minutes. The sensation of being unable to quite arrive, of thinking arriving in pieces, of the ordinary world having moved somewhere further away than the few seconds of being asleep should explain. This is the risk of the nap that extends too long, not the sleep itself, but the particular quality of waking from it. But a nap that runs longer still, longer than 45 minutes, approaching the full span of a complete natural sleep cycle, begins the slow passage out of slow wave sleep through its own internal architecture, ascending through lighter stages towards something that could not be predicted from a brief nap surface behavior alone. A full 90 minutes is approximately the duration of one complete ultradian sleep cycle. The first portion, from sleep onset, passes through stage 1 into stage 2, and then descends into stage 3, slow wave sleep. This descent typically takes between 20 and 40 minutes from the moment of sleep onset, depending on the accumulated sleep pressure and the circadian phase at nap time. The brain spends some portion of the middle of the cycle in slow wave sleep, and then, if undisturbed, it begins to ascend. The delta waves diminish in amplitude and frequency. Stage 2 reemerges, the spindles returning in their familiar waxing and waning pattern, signaling that the deepest part of the cycle has ended and the architecture is climbing back toward lighter sleep. And then, at the end of the ascending arc, something appears that has no equivalent in the earlier stages of the cycle, and no obvious resemblance to anything the sleeping brain has been doing in the 40 or 50 minutes before. REM sleep. The EEG during REM is by every superficial appearance close to waking. The slow sweeping delta waves are entirely gone. The spindles that marked stage 2 are absent. What replaces them is a low amplitude, high frequency, desynchronized pattern almost identical to the EEG of an alert, awake person with eyes open. The brain, in this final phase of the 90-minute cycle, is electrically active in a way that more closely resembles wakefulness than it resembles the sleep it has just emerged from. And yet the body, during REM, is more profoundly paralyzed than at any other point in sleep. The major voluntary muscles, the limbs, the torso, are actively inhibited, held still by a mechanism originating in the brainstem that prevents the motor commands generated by the dreaming brain from being executed. The cortex is firing, the body is not moving, the eyes are an exception. Beneath the closed lids, the eyes move in rapid conjugate bursts, small, quick, purposeful seeming rotations that give this state its name, recorded by electroculography. Electrodes placed near the outer corners of the eyes. These movements appear on the trace as small voltage deflections, brief and irregular, punctuating the record in clusters separated by intervals of stillness. What these movements correspond to in the interior experience of the dreaming brain has been a subject of long discussion, but the movements themselves are precisely real, mechanically distinct from the slow drifting eye movements of stage 1 or the stillness of slow-wave sleep, and visible in the record as a clear and repeating signature. What is happening in the brain during REM has a distinct neurochemical character as well as a distinct electrical one. The brainstem nuclei that release norepinephrine and serotonin, the locus coeruleus, and the dorsal raphe nucleus, structures that during waking maintain a continuous background tone of arousal, fall nearly silent during REM sleep. Their activity, which has been running as a steady background current through every waking hour and has sustained, even at reduced levels, through NREM sleep, drops to its lowest point in the sleep cycle. In their place, acetylcholine release from the basal forebrain rises sharply, and this shift in neurochemical balance, high acetylcholine, minimal norepinephrine, minimal serotonin, creates a brain state that is electrically active and chemically unusual, a state for which waking provides no close equivalent. This chemical landscape does specific things. High acetylcholine combined with the suppression of the norepinephrine and serotonin background creates conditions under which associative links between distant pieces of stored information become more accessible. During waking, the norepinephrine tone tends to keep thought directed and coherent, holding attention to immediate tasks and filtering out information not directly relevant to the current goal. During REM, with that tone removed, the brain's activity spreads more widely, following links between memories that are distant from one another by ordinary waking logic, emotional associations, thematic connections, metaphorical resemblances, rather than strict categorical ones. A content of dreams, with its characteristic quality of things being adjacent that have no ordinary reason to be together, is partly a reflection of this altered associative landscape, a brain that is fully active but navigating by different rules. A longer nap that reaches REM sleep arrives at the end of its cycle in this state and wakes from it. Emerging from REM, unlike emerging from slow-wave sleep, is a relatively gentle process. The brain was already running near waking levels of activity. The neurochemical shift from REM's acetylcholine dominance back toward the norepinephrine and serotonin tone of waking is a matter of the brainstem arousal nuclei resuming their normal firing, which they do relatively quickly. The subjective experience of waking from REM is typically closer to a clear emergence than to the confused groping of waking from slow-wave sleep. The cycle completes, the brain surfaces. The person is, by most subjective measures, more refreshed than they would have been after the same duration of sleep interrupted in the middle of slow-wave sleep. The difference in how they wake as real and consistent as the difference in the stages from which they wake. This is the significance of the cycle's length. 90 minutes is not an arbitrary number or a convenient round figure. It is approximately the natural duration of the brain's own sleep architecture before it returns to the surface. A nap of this length follows the brain's internal trajectory rather than interrupting it, and the place it ends, in REM close to waking, the cortex already running near its alert frequency, means the transition back to the waking world requires the shortest crossing. The spindle-rich stage 2 of a short nap, the slow oscillating depth of stage 3, the electrically near-waking activation of REM, these are not arbitrary labels applied to different lengths of sleep. They are genuinely different brain states, with different cellular mechanisms, different neurochemical signatures, and different relations to the waking and sleeping that surround them. A 10-minute nap and a 90-minute nap share a name and share the broad condition of a person lying still, with eyes closed, but what the brain is doing in each bears almost no resemblance to what it is doing in the other. Sleep spindles To follow one thread a little further down into the structure of what stage 2 produces, are not simply markers of sleep depth. They are instances of a specific organized neural computation, the product of that thalamocortical loop, the reticular nucleus firing in synchronized bursts, and the relay neurons carrying that rhythm upward to the cortex. In the short nap that reaches stage 2, 20 or 30 or 40 of these events accumulate across the record, each one brief, each one real. The thalamic reticular nucleus, thin and curved, running like a shell around the outer surface of the thalamus, coordinates each one. Its inhibitory neurons synchronize. The rhythm propagates inward. The cortex receives it, rises briefly, and then settles, and the trace at the scalp shows a spindle, a small organized shape against the background, waxing and waning over its second or two of existence, then gone. The cortical slow oscillations of stage 3 are generated differently and in a different place. Their origin is in the cortex itself, in the intrinsic membrane properties of cortical neurons that, when released from the irregular stimulus-driven demands of waking, fall into a coordinated rhythm. The downstate, in which neurons go quiet, is not forced from outside. It arises from within the population, from a collective transition into hyperpolarization that spreads across the cortex as each neuron's silence contributes to the silence of its neighbors. The upstate, in which neurons fire together, arises from the rebound from this collective quiet. The oscillation sustains itself through the network's own properties, with no external clock required. The delta wave in the EEG is the scalp-recorded summing of this cortical rhythm, its amplitude high because so many neurons are participating simultaneously, its frequency slow because each full cycle, downstate to upstate and back, takes the better part of a second. These waves appear in the nap at the 30-minute mark, in the 40-minute mark, deep and wide in the spectrogram, showing as broad bands of warm color in the slow frequency range. And they persist for the napper who is allowed to sleep through them until the cycle's homeostatic machinery begins to draw the architecture back upward, the slow wave pressure partly discharged, the brain gradually releasing its grip on the deep oscillation, the delta power declining in the record as the spindles of stage 2 return and the approach toward REM begins. REM, when it arrives, appears in the EEG as an abrupt change in everything. The slow waves that had dominated are entirely gone. The spindles that had returned briefly in the ascending stage 2 also disappear. What takes their place is a low amplitude, mixed frequency record that looks to every naive reading like waking, the desynchronized fast signal. The active brain, running without the deep coordination of NREM, running instead in the spread and associative mode that makes this state unlike any other. A small voltage recorded near the outer corner of an eye, a brief deflection to the right, then to the left, then stillness. The eyes have moved, the brain is dreaming, the body has not moved at all. The limbs are held by a mechanism that originates in the medulla and the pons, brainstem structures that during REM send an active inhibitory signal through the spinal cord to the motor neurons, suppressing the muscle tone that would otherwise allow the commands being generated in the motor cortex to reach the body. The sleeping person may be running, or reaching, or speaking in the context of the dream. None of it arrives at the muscles. The record shows complete stillness externally, and the active high-frequency near-waking EEG internally, and between them a gap that is one of the defining features of this state. This is the brain that a 90-minute nap ends in. Cortex active, eyes moving, body still, norepinephrine and serotonin at their lowest point, acetylcholine elevated, associations flowing, without the directed filtering of waking. And then, for the napper who is allowed to complete the cycle and wake naturally from this state, a transition back to waking that requires the shortest crossing. The cortex is already most of the way there. The brainstem nuclei need only resume their normal firing. The motor inhibition releases, a breath deepens, or a sound from outside crosses the now lowered threshold, and the eyes open into a room that feels available, close, and clear. What a short nap produces, the 20 or 30 or 40 spindle events accumulated in stage 2 during 10 or 15 minutes of sleep, is genuine and distinct from rest alone. What a full cycle produces, the slow wave passage of stage 3 and the activated associative passage of REM, is more elaborate, more complete in its architecture, more closely resembling what a full night of sleep achieves on a smaller scale. Both are real, both are different, and both are products of the same system, the same hierarchy of oscillations and transitions and neurochemical states that the sleeping brain has been cycling through for as long as there have been mammals, long before anyone thought to place an electrode on a scalp and watch the waves. The transition from stage 3 back toward REM within the full 90-minute cycle is not abrupt. It is a slow ascent, taking its own several minutes, the delta waves diminishing gradually rather than cutting off, their amplitude shortening and their frequency increasing in small steps, the spectrogram showing the warm high amplitude slow wave power fading at the left edge, while the sigma band color of spindle activity begins to re-emerge toward the right. This is the brain's own pacing. No external mechanism drives the transition. The homeostatic discharge of slow wave sleep, the partial clearing of the accumulated sleep pressure that drove the descent into stage 3, releases the architecture to begin climbing, and it climbs at its own rate, in its own time, following the internal logic of its own oscillatory systems. A stage 2 spindle appearing in this ascending portion of the cycle looks, on the trace, identical to a spindle appearing in the initial descent through stage 2 40 minutes earlier. The thalamic reticular nucleus generates it by the same mechanism. The thalamocortical loop sustains it by the same interaction, and yet this spindle is occurring in a different context, after the brain has passed through slow wave sleep, after the homeostatic pressure has been partly discharged, on the way up rather than on the way down. The same mechanism, the same shape on the trace, a different location in the architecture, and a different relationship to what comes before and after it. This is the ordinary complexity of sleep in an afternoon nap. Not a simple dimming of consciousness, not a uniform reduction in brain activity, but a structured progression through states that are genuinely different from one another, and that succeed one another in a consistent order for reasons rooted deep in the biology of the brain. Stage 1 as the corridor, stage 2 with its spindles and its K-complexes, the thalamic rhythm sustaining itself in its brief repeated loops. Stage 3 with its vast, slow, synchronized oscillation spreading across the cortex in wide delta waves, each one a second of coordinated silence, followed by a second of coordinated firing. The long middle passage of the cycle, and then the ascent, the return of spindles, the approach toward REM, and finally, REM itself, the cortex alive with near-waking activation, the eyes making their rapid conjugate movements beneath closed lids, the body held completely still, the brain running in its most unusual chemical state. This is what is happening behind closed eyes, in a quiet room, on an ordinary afternoon, in a brain that has found the conditions to complete its own natural cycle. The sleeper is not aware of any of it. The transitions between stages produce no felt sensation of crossing from one territory to another. There is only the receding of the waking world, a time that cannot be measured from inside, and then the gradual or sudden return of it. But the transitions are real, the stages are real, the spindles accumulate on the record, the delta waves sweep across the cortex in their slow, wide arcs. The REM brain activates in near silence, and at the end of a full cycle, when the napper wakes from that activated dreaming state, the cortex is already lit with the near waking signal. The body is releasing from its held stillness. The first breath of waking, deepening slightly in the chest, and the brain that opens its eyes is a brain that has completed something, that has followed its own internal arc from beginning to surface, having passed through every stage the cycle contains, and having arrived, by its own route, at the edge of the world again. The room is the same room, the light is unchanged, the sound from outside is continuous, and behind closed eyes, in the seconds before waking, the brain has been somewhere the waking world cannot reach, somewhere structured and orderly and deep, organized around oscillations that have been running in sleeping animals for longer than our species has existed, ancient rhythms cycling beneath the surface of ordinary rest, doing their quiet work in the dark. Deep in the tissue of the brain, tucked into the inner curve of the temporal lobe, the hippocampus had been working since early morning. It had been receiving the day, each face, each word, each turning of an unfamiliar corner, and encoding what arrived into patterns of electrical activity that might in time become memory. By late afternoon, something in that encoding had begun to slow, not because the world had grown less interesting or because attention had wandered, but because the cellular machinery of memory had approached its limit, the way a vessel fills, not suddenly, but by degrees, each new addition displacing a little more of the space that remained. Matthew Walker and his colleagues at the University of California, Berkeley, measured this process directly. In 2010, they scanned the brains of people learning new material at different points across a long waking day, and what appeared in the imaging data was specific and spatially precise. The hippocampus, bright with activation during morning learning sessions, showed measurably reduced engagement by late afternoon when subjects attempted to encode new information. The signal had not vanished. It had dimmed. The same region, the same subjects, hours later, working harder for a smaller return. The brain had not stopped trying. It had simply grown less able to take in what the world kept offering. That diminishment had a name and a mechanism. It was not fatigue in any loose or general sense. It was a measurable change in the functional state of a specific structure, brought on by the continuous demands of a long, unbroken day. And when the researchers gave one group of subjects a 90-minute nap in the early afternoon, a nap with a full ultradian arc, moving through the stages of sleep in order, that group came back to the learning task with hippocampal activation that had not merely recovered, but had risen above even the morning's levels. The nap had not simply restored what the afternoon had taken. It had returned the brain to a state better than the one it had occupied at the start of the day. The hippocampus held the story of how this was possible. It generated during slow-wave sleep a particular kind of electrical event, brief, rapid, appearing and disappearing in a fraction of a second. When recorded with electrodes placed near the structure itself, these events appeared as sudden bursts of high-frequency oscillation, distinct against the slower background of sleeping brain activity, each one lasting perhaps a tenth of a second or less. The researchers who first described them named them sharp-wave ripples for the shape they left in the electrical record. They oscillated somewhere between 80 and 100 cycles per second, far faster than the slow rhythms of deep sleep that surrounded them, and they seemed to carry within their brief duration something organized and specific, not random noise, but a pattern. What the pattern contained was the day's experience, compressed and replayed. The neural populations that had fired during waking learning fired again, in the same sequence, in a fraction of the original time. A walk through a new room, encoded over minutes, could be replayed in the hippocampus during sleep in something closer to a heartbeat. The sharp wave ripples were the mechanism by which recent experience traveled inward and downward, from short-term storage towards something more durable. Each replay was a step in the process of deciding, without deliberation, what was worth keeping. They did not work alone. The ripples arrived embedded within a slower rhythm, the upstate of a cortical slow oscillation, a wave of neural activity that swept across the brain's surface roughly once per second during the deepest stage of sleep, its high amplitude rise visible on a recording as something that dwarfed everything around it. The slow oscillation opened a window. The sharp wave ripples arrived inside it, and threaded through both, moving at a speed between the two, came the sleep spindles, 12 to 15 cycles per second, generated in the thalamus and spreading outward through the cortex in waxing and waning bursts, each one lasting between half a second and 3 seconds. Each one a coordinated event in the thalamocortical network that linked the brain's deeper relay station to its outer surface. The three rhythms nested inside one another, each occupying a different timescale, each arriving in a sequence that was not random. The slow oscillation set the tempo. The spindles clustered at the peak of each slow oscillation's upstate. The hippocampal ripples clustered within the spindles. A memory trace, compressed and electrical, traveled up through this nested sequence and found its way toward the cortex, toward the more permanent storage that would outlast the sleep and be accessible days or months or years later. The hippocampus was not simply filing information away. It was broadcasting, and the cortex, synchronized and receptive within the architecture of slow-wave sleep, was receiving. This mechanism, the three-way coupling of slow oscillations, spindles, and ripples, explained in cellular terms why the hippocampus felt emptied out by the end of a long day and restored after sleep. Memory consolidation was not a passive process of things being held in place. It was an active transfer running at full speed across the sleeping brain, moving the contents of a temporary store into a more distributed and durable form, leaving the hippocampus cleared and ready for the next day's intake. The nap had not merely rested the brain. It had performed a specific operation on the memory system, one that no amount of quiet wakefulness could replicate. The improvement Walker's team measured was not slight. Subjects who had napped showed a 20% improvement in the capacity to encode new material compared to those who had remained awake across the afternoon. 20% is a large difference in a cognitive domain. It is the difference between a day that accumulates and a day that increasingly fails to do so. The researchers also found that the magnitude of benefit tracked not simply with nap duration, but with a more specific measure, the density of sleep spindles during stage 2 of the nap. The more spindles, the greater the restoration of hippocampal capacity. The mechanism and the outcome matched precisely, each one pointing to the other. A room at rest in the early afternoon, with curtains drawn and a person lying still inside it. From the outside there is almost nothing to observe. The breath slows, the face relaxes, nothing appears to be happening. But from inside the sleeping brain, in the cellular terms the researchers spent years learning to read, an enormous amount is underway. Sharp wave ripples fire in rapid succession in the hippocampus. Spindles arrive in the thalamus, spread outward, cluster at the peaks of slow oscillations. Memory traces are carried from one system to another in a coordinated relay that has been running in sleeping animals since long before there were people to study it. The quiet of the room is entirely misleading. Attention is different from memory, and a nap reaches it by a different path. The brain circuits responsible for sustained attention, the ones that maintain the constant low-level vigilance required to catch an unexpected event, notice a change, respond to something unexpected, depend on a different set of neurochemical conditions. They are sensitive to adenosine accumulation, which builds across the waking hours and blunts the responsiveness of neurons in the basal forebrain that use acetylcholine as their chemical messenger. Those cholinergic neurons project widely through the cortex, maintaining the state of broad readiness that attention requires. When adenosine levels rise, the cholinergic signal weakens and the readiness falters. During even a short nap, 10 minutes or 20, confined mostly to the lighter stages of sleep, adenosine is cleared from the basal forebrain at a rate that wakefulness cannot achieve. The clearance is not complete across such a brief sleep, but it does not need to be. Enough adenosine is removed to restore a measurable degree of cholinergic tone, to raise the readiness of the networks that sustained attention depends on, to push response times back toward the sharp and precise end of their range. The nap is not undoing hours of accumulation in a few minutes of sleep. It is moving the needle by enough to matter. The psychomotor vigilance task was designed specifically to measure this kind of attention, sustained, effortful, continuous. A subject sits before a small device and presses a button whenever a light appears. The light appears at unpredictable intervals. The task requires no intelligence, no strategy, no particular knowledge. It requires only that the brain stay ready for as long as the test runs and respond without delay when the stimulus arrives. Response times are recorded to the millisecond. Lapses, responses that take longer than half a second, are counted separately because a half-second delay in a real environment can be the difference between catching something and missing it entirely. Sleep-deprived subjects performing this task before a nap show a characteristic pattern in their data. Most responses are reasonably quick, but scattered through the distribution are outliers. Responses that took 800 milliseconds, a full second, sometimes longer. These are the lapses, the moments when the brain's readiness dropped below the threshold required to respond at all, and the stimulus appeared in that window. The raw data for a fatigued person looks ragged at the edges. The peak of the distribution is still near the median, but the tail extends far to the right, a visible sign that something is failing intermittently. These are not errors of skill, they are failures of the most basic alertness. After a short nap, 15 minutes, 20, the same subjects return to the task, and the distribution changes. The peak stays in roughly the same place. It is the tail that shortens. The lapses, those long outliers extending into the seconds, nearly disappear. The brain has not become faster in any general sense. The fast responses were already fast. What has recovered is the floor, the maintenance of a minimum level of readiness across the whole duration of the task, the suppression of the gaps in attention that accumulate when adenosine has been rising unchecked for too many hours. The nap has repaired the bottom of the distribution, and in doing so has made the whole record look like someone who slept. This recovery in sustained attention is not subtle or ambiguous in the laboratory data. It is statistically significant, operationally meaningful, and it appears consistently across subjects and studies. The neurophysiological account underneath the behavioral change points to two convergent processes: the restoration of norepinephrine tone in the locus coeruleus, a small brainstem nucleus whose chemical output is essential for the kind of broad cortical arousal that underpins vigilance, and the partial reversal of adenosine-mediated inhibition in the cholinergic basal forebrain. Both processes run simultaneously during even a brief sleep. Both contribute to the recovery of attention, and neither can be reproduced by rest alone. Lying quietly in a dark room with eyes closed, without sleep, produces a much smaller and shorter-lived improvement on the same task. The locus coeruleus sits deep in the brainstem, small enough that the entire nucleus fits within a few millimeters of tissue, pale blue in the gross dissection of a human brain. Its name means the blue place. From that small cluster, norepinephrine-releasing fibers project throughout the brain, reaching the cortex, the thalamus, the limbic system, the spinal cord. When the locus coeruleus fires, its chemical output spreads broadly, raising the signal-to-noise ratio across large networks, sharpening the response to meaningful stimuli relative to background. When adenosine accumulation depresses its activity across a long waking day, the whole brain feels the dimming. When a nap restores norepinephrine tone, even partially, the brightening is similarly broad. Motor learning follows a path through the sleeping brain that is distinct from declarative memory, but equally well traveled. A new physical skill, a sequence of finger movements, a technique practiced in training, encodes differently than a fact or a name. The motor sequence lives in the sensoromotor cortex, in the cerebellum and basal ganglia, in the circuits that govern precision and timing, and the smooth flow from one movement to the next. And like the hippocampus in declarative learning, these circuits are not simply passive repositories. They continue working after the learning session ends, and sleep provides the condition under which that offline work runs most efficiently. Walker and his colleagues demonstrated this in a study of motor sequence learning. Subjects learned a finger tapping sequence, a specific pattern tapped out in order, as quickly and as accurately as possible. After training, one group spent the following night asleep, while another did not. At the test session, those who had slept showed performance gains that were substantial, their speed and accuracy having improved between the training and the test, not because they had practiced more, but because sleep had continued the work. More relevantly for nap research, subjects who napped after training on the sequence showed performance gains at an afternoon test session that closely matched what the overnight sleeping group achieved at their evening test. The nap had done what the full night had done on a compressed time scale for this particular kind of learning. The fMRI data from these sleeping nappers gave the finding a physical location. During the nap, as subjects moved through the stages of sleep, the motor cortex showed task-related activation, the same spatial pattern that had appeared during waking practice, now recurring in the quiet, sleeping brain. The sequence was being rehearsed again, without hands, without any movement visible from the outside, in the neural territory where the original learning had taken place. The magnitude of this reactivation during the nap predicted the size of the performance gain measured afterward. The brains that replayed the sequence most vigorously during sleep improved the most at test. Offline consolidation was not a uniform process happening equally in every sleeping brain. It was proportional, and the imaging made that proportion legible. There is something in the image of a sleeping person's motor cortex lighting with the pattern of a task they have not performed in hours, a task they are not consciously thinking of, in a brain where consciousness itself is suspended, that changes how the quiet of sleep looks. The stillness is the outside of an interior activity that has its own intensity, its own order, its own directed motion through the neural architecture. The brain during sleep is not switched off. It is switched to something else, a mode of processing that waking cannot replicate, in which the gains of practice are sorted and deepened and readied for use. A flight is long, and the cruise phase, once established, can run for hours across open water or empty continent. Inside the cockpit, the pilots monitor instruments whose needles barely move, respond to occasional communications, watch a world that from this altitude is slow and featureless. The physiological cost of this sustained attention across hours is real and measurable. Response times lengthen. Microsleeps appear in the EEG record, brief intrusions of sleep-like brain activity into a waking record, lasting seconds, often completely unnoticed by the person experiencing them. A microsleep is not ordinary drowsiness. It is a window opening and closing in the neural record, during which the brain is by every electrical measure briefly asleep. In the 1990s, researchers at the NASA Ames Research Center, led by Mark Rosekind, studied this directly in the operational environment of actual transoceanic flights. Pilots were given a scheduled rest opportunity during the cruise phase, 40 minutes set aside for sleep, with the full understanding that some of that time would be occupied by lying down rather than sleeping. The actual sleep time, measured by EEG, averaged about 26 minutes. Not a long nap, not a deep one. Constrained by the environment, by the difficulty of sleeping in a cockpit seat, by the residual alertness of people who rarely had occasion to nap on the job. 26 minutes of real sleep, embedded in 40 minutes of rest. The comparison was made against a group of pilots who had no scheduled rest opportunity during the same flight phase, who remained awake and on watch continuously. At the approach and landing phase, the most demanding period of any flight, requiring the highest levels of sustained attention, rapid response to changing conditions, and precise decision-making, the rested group showed a 34% reduction in physiological microsleeps compared to the control group. Their reaction times improved by 16%. These were not small effects in a laboratory setting with graduate students pressing buttons. These were operational differences in working pilots, in real aircraft, measured during the phase of flight, where the margin for error is narrowest. The EEG traces from that study made the stakes of the finding visible in a way that numbers alone could not. A cockpit EEG recorded during approach showed in a pilot who had not rested brief intrusions of delta and theta activity into the waking record, sudden shifts in the frequency pattern lasting 5 or 6 seconds, appearing without warning and disappearing just as abruptly. Each intrusion marked a moment when the pilot's brain had slipped into a state functionally indistinguishable from sleep. The hands remained on the controls. The posture did not change. From outside, nothing visible had occurred, but the brain's electrical signature had traveled, briefly and involuntarily, somewhere else, and during those seconds, the instrument panel in front of a sleeping brain was receiving no useful processing. In the pilots who had napped, who had slept for 26 minutes on average in a seat during the cruise, those intrusions were fewer and shorter. The waking record was cleaner. The approach was managed by a brain that had moved its adenosine needle, even a modest distance, in the direction of restored readiness. 26 minutes of sleep had produced a 34% reduction in the brief involuntary absences that sleep deprivation makes inevitable. The numbers were large because the baseline was poor and the task was unforgiving, but the mechanism was the same one that appears in every laboratory measure of nap-related attentional recovery. Adenosine, partially cleared, norepinephrine tone, partially restored. The floor of sustained attention lifted to a level that waking rest alone cannot reach. The findings from that program of research moved napping from a personal habit into an operational consideration, one that aviation authorities, military planners, and shift schedulers began to take seriously. But what is true of pilots under sustained operational demand is simply a more visible version of what is true of any brain that has been working for hours without rest. The microsleeps that appeared in the cockpit EEG are the same phenomenon at the same time scale that appears in the data of a student sitting at a desk in the late afternoon, or a driver on a familiar road whose miles suddenly can't be accounted for, or anyone whose waking day has been long and whose adenosine has been rising since before the morning coffee had fully taken hold. The gap between a rested and an unrested brain is not always visible from the outside. Behavior looks nearly the same. Conversation continues. Tasks are undertaken with apparent effort, but in the electrical record, in the response time distributions, in the spatial pattern of hippocampal activation during learning, the difference is legible, specific, and large. The nap does not erase that gap completely, but across the measures where the gap appears, a short sleep narrows it in ways that rest without sleep does not. The benefits of a nap are not distributed equally across all cognitive functions. The gains are most pronounced in the domains most sensitive to adenosine accumulation and most dependent on the sleep-active restorative processes. Sustained attention, the kind that falters first when adenosine rises, recovers with even a short nap. Declarative memory encoding, which depends on hippocampal capacity that fills and needs to be cleared, recovers best with a nap long enough to include substantial slow-wave sleep and its associated spindle activity. Motor learning consolidation runs most efficiently when the nap includes both slow-wave and REM components. Each additional stage of sleep adds something specific. The relationship between nap duration and the specific cognitive function being restored is not arbitrary. It reflects the precise requirements of each process at the level of biology. The attentional system needs adenosine cleared, and that process begins quickly, within minutes of sleep onset. Clearance is underway, and 20 minutes of sleep moves the needle by a meaningful amount. The declarative memory system needs spindle activity, which arrives reliably in stage 2 and accumulates in proportion to the time spent there. The motor consolidation system benefits from REM, which arrives reliably only after the earlier stages have run their course. Different recovery processes have different timescales because they depend on different stages, and different stages arrive in a fixed order that cannot be skipped. This is why the question of optimal nap length has no single answer, and why attempts to give one produce the frustration of advice that works for some functions and not others. A 10-minute nap restores attention. It does not substantially restore hippocampal encoding capacity. A 60-minute nap restores hippocampal capacity and deepens motor consolidation. It also carries the risk of waking from slow-wave sleep into a period of disorientation that temporarily reverses some of its gains. A 90-minute nap completes a full cycle, reaches REM, and provides the broadest cognitive restoration at the cost of a commitment in time that most afternoons cannot easily accommodate. The biology is not flexible on these parameters. The stages arrive when they arrive, and their benefits accumulate in proportion to the time spent inside them. Across a long waking day, the brain's internal architecture does not degrade uniformly. It degrades in proportion to demand in the specific circuits and systems that have been called on most heavily, and in the general condition of readiness that depends on the neurochemical state of the sleeping brain system. The hippocampus dims toward evening not because learning becomes less possible in principle, but because a specific functional capacity, the ability to form new associations in a system already carrying a full load, has been gradually exhausted by the morning's and afternoon's intake. The nap interrupts this trajectory, not by resetting everything, not by returning the brain to the state it occupied at sunrise, but by performing a targeted restoration across the specific systems most in need of it. For the attention system, that restoration is the partial clearance of adenosine and the recovery of a neurochemical readiness that wakefulness continuously consumes. For the memory system, it is the active transfer of recent experience out of the hippocampus and toward more durable cortical storage, leaving the temporary repository cleared and available for the next round of encoding. For the motor system, it is the silent rehearsal of practiced sequences in the neural territory where they were first laid down, the sleeping repetition of a skill without any physical movement at all. The finding from Walker's 2010 study that the hippocampus could be restored to a state better than its morning baseline is worth sitting with because it exceeds the intuitive expectation. A restored baseline is a good outcome. A restored baseline that exceeds the starting point is a different kind of claim. The mechanism is not entirely settled, but the most plausible account involves the active nature of memory consolidation during sleep, the sharp wave ripples, the spindles, the slow oscillations running their coordinated relay, which does not simply stop the accumulation of memory load, but actively reduces it, freeing the hippocampus from material it has been carrying and making room that was not available before the sleep began. The nap does not merely pause the filling of the vessel, it empties some of what had collected. A brain that has run all day on unbroken wakefulness, measured by every available laboratory instrument as afternoon approaches, is not the same as a brain that had a short sleep in the early hours of the afternoon. The EEG traces are different. The response time distributions are different. The hippocampal activation during new encoding is different. The microsleep rate is different. These are not subtle differences at the edge of detectability. They are large, consistent, and replicable across decades of research using different measures, different populations, different nap durations, different laboratory environments. The picture is not ambiguous. It is a picture of two different physiological states, one of which learns and responds and attends more efficiently than the other. The pilot on approach, managing an aircraft in the final minutes before landing, whose EEG shows a clean and continuous waking record, is in a different functional state than the pilot whose record shows those brief, involuntary dips. The student whose hippocampal activation is bright and robust during an afternoon learning session is processing new material in a different condition than the one whose activation has dimmed to a fraction of its morning level. The laboratory subject, whose response time distribution shows no tail of lapses, is sustaining attention in a way that the unrested subject cannot. These differences were produced in each case by roughly 20 to 90 minutes of sleep taken in the middle of the day. The value of that sleep does not lie in its length. It lies in what the sleeping brain does during it, the chemistry it reverses, the electrical events it generates, the processes it runs that have no waking equivalent. The quiet of a nap is not empty. It is full of activity that cannot be consciously chosen or directed or replicated by will. It runs on its own, according to its own order, in the neural architecture that every sleeping animal has carried for longer than any of the functions it now serves have existed in their current form. The cockpit EEG trace showing a pilot's waking record, clean, continuous, desynchronized, the mark of a brain staying present in the demands of the moment, and the same trace from a pilot who had not rested, those brief intrusions of delta and theta activity, each one a window through which the instrument panel was briefly unmonitored, together make the argument that no verbal description quite achieves. One brain was there, the other was, for seconds at a time, somewhere else. The nap had returned the first pilot from somewhere else back to the cockpit, not by magic and not by discipline, but by the ordinary and ancient and precise process of sleep, doing what sleep has always done, clearing the chemical accumulation of wakefulness, rehearsing what was learned, restoring the readiness that the waking day continuously spends. Outside the window of the research laboratory, and outside the cockpit, and outside any setting in which the measurement instruments have been installed, this same process runs every afternoon in every brain that finds a short sleep. The EEG is not there to read the spindles as they form. The fMRI is not there to show the hippocampus brightening again as the nap ends, but the activity is underway regardless. In the dark behind closed eyes, in the slow electrical architecture of a sleeping brain that has been doing this for a very long time and needs no witness to do it well. There is a particular kind of waking that does not feel like waking at all. The room is dim, the body is on a couch or a bed, the eyes have been closed for what felt like minutes, and then something breaks through. A door, a sound, the slow sense of obligation returning, and what comes back is not alertness, but something murkier, slower, heavier than sleep itself. The hands move, the eyes open, but the mind does not follow. It is still somewhere else, still wrapped in something that has not finished, and the commands sent from the brain to the body arrive late, as if crossing a very long distance. This is a well-known feeling. It has a name and a mechanism and a duration that can be measured. The brain, in that moment, is not simply waking up. It is waking up from the wrong place. And the distinction between a depth of sleep the body had not finished with and a lighter layer from which emergence is clean and quick is one of the most consequential things about a short sleep, more consequential in some ways than whether the nap happened at all. The name for this state is sleep inertia. The feeling is familiar to almost anyone who has ever slept too long in the afternoon, but the mechanism is specific, and the brain it happens inside is doing something measurable and distinct. The EEG trace of a person pulled out of stage 3 NREM slow wave sleep shows something that should not be there. The waking record, low amplitude, fast, desynchronized, is interrupted. Moving through it, still present and still visible, are the tall, slow signatures of the deepest sleep. Delta waves. They rise and fall over 1 to 2 full seconds. Their amplitude dwarfs everything around them. In a waking record, they look like intrusions, like transmissions from a station that was supposed to go quiet. The person is sitting up, the eyes are open, the delta waves are still running, the brain has not fully woken, not in the parts that matter most. The mechanism begins in the structure of deep NREM sleep itself. As sleep deepens past the lighter stages, past the sleep spindles and the K-complexes of stage 2, the cortical EEG begins to fill with slow, high-amplitude delta oscillations at frequencies between half a cycle and 4 cycles per second. The neurons of the cortex are synchronizing in vast coordinated waves. Hundreds of millions of cells briefly fire together in what is called an up state, then fall silent together in a down state, then fire again. This alternating rhythm, sweeping across the cortex in slow pulses, is the electrical signature of the brain's deepest recuperative state. It is the architecture of slow-wave sleep, and when a nap ends inside it, rather than before or after it, The brain's return to wakefulness is not a clean transition, but a slow, incomplete one. What makes it incomplete is the prefrontal cortex. Not all regions of the brain wake at the same rate. The brainstem comes back first, and quickly the arousal systems there, the structures that control the body's basic alertness, reactivate within seconds or minutes. Primary sensory and motor cortices follow, bringing perception and movement back online. But the prefrontal cortex, sitting at the forward edge of the brain and carrying the full weight of executive function, decision-making, inhibitory control, working memory, the ability to hold competing possibilities in mind, is the last to come back. And in fMRI images taken in the minutes after a person wakes from slow-wave sleep, the brainstem and thalamus are already bright and active, while the prefrontal cortex remains visibly dim, its activation still incomplete, still lagging, still below the level it needs to operate clearly. The brain, in those images, is a portrait of two states occupying one space. The person is awake in some regions and still asleep in others. The parts that first return are the ones that allow the body to sit up, to hold a cup, to answer a question in a way that sounds approximately coherent. The parts that have not returned are the ones responsible for knowing whether the answer is actually right. Working memory remains compressed. Reaction time is slower than waking, but not slow enough to feel wrong from the inside. The sense that the mind has returned to full function arrives well before the function itself does. This gap between the subjective feeling of alertness and the objective presence of it is one of the more important features of sleep inertia, because it means the state can be invisible to the person experiencing it. The brain wakes from the outside in. The last layer to return is the layer doing the thinking. The prefrontal cortex, after awakening from slow-wave sleep, can show elevated delta wave power above its waking baseline for anywhere between 5 and 30 minutes. The range is real. Some people emerge quickly. Others carry the signature for half an hour, sitting at a desk or behind a wheel or in a conversation, performing tasks with a brain that is still, by one precise measure, partly asleep. The delta waves are not felt as drowsiness, or not entirely. They are felt as a kind of cognitive friction, a thickening of thought, a delay between intention and execution, a sense that the mind is working harder than it should be for the difficulty of what it is doing. The cognitive reactivation follows a spatial sequence, and that sequence is not random. Subcortical arousal structures first, primary cortices second, association cortices third, prefrontal and anterior cingulate cortices last. The anterior cingulate cortex, which monitors for conflict, adjusts behavior, flags errors, comes back alongside the prefrontal cortex, which means the brain's ability to detect its own mistakes is one of the last faculties to return. The person does not notice they are performing poorly. The noticing mechanism is still reactivating. This is not a small thing in practical terms. A driving simulator study of subjects woken from slow-wave sleep shows what this looks like, expressed as physical behavior. The steering corrections are slower. They are more erratic. They arrive later relative to the road condition that prompted them, as though processed through a slight but consistent delay. They are less anticipatory. The driver responds to what has already happened rather than what is about to happen. Compared to the same person tested after waking from stage 2 NREM, the difference is visible in the smoothness of the plotted steering line. One curve flows, adjusting ahead of events. The other catches up, corrects, overshoots slightly, corrects again. The lag is not large, but on a real road, at highway speed, it is not nothing. What drives a nap into slow-wave sleep, rather than keeping it in the lighter stages, is a function of two variables working together. The first is homeostatic sleep pressure, the accumulated chemical weight of wakefulness, rising with every hour the brain has been running. The greater the pressure at the moment the nap begins, the faster and deeper sleep descends. A person who has been awake for 9 hours and lies down in the early afternoon carries substantially more sleep pressure than a person who has been awake for 6. The brain of the 9-hour waker enters slow-wave sleep more quickly, reaches it more completely, and holds it longer. The brain of the 6-hour waker may stay in the lighter stages, where the spindles live, and the delta waves have not yet taken hold. The second variable is circadian phase. The circadian alerting signal from the suprachiasmatic nucleus follows a curve across the day, and the point in that curve at which a nap begins determines how much resistance the circadian system offers against the pull toward deep sleep. Early in the afternoon, the circadian alerting signal is still relatively robust, holding against the homeostatic pressure. Later in the afternoon, after 3 in the afternoon, the circadian signal has not yet begun its evening rise, and the alerting support is at a low ebb. Homeostatic pressure and circadian phase both pull toward depth at that hour, and depth means slow-wave sleep, and slow-wave sleep means the risk of waking from the wrong place. The practical consequence is visible in EEG spectrograms recorded at different nap times. A nap begun at 1 in the afternoon in the same person on the same day enters slow-wave sleep after roughly 25 minutes of sleep. A nap begun at 3 in the afternoon enters slow-wave sleep in perhaps 12. The spectrogram of the later nap shows the warm, high-amplitude colors of delta power spreading across the frequency plot more quickly from sleep onset, not because anything has changed about the person, but because two forces are pulling in the same direction at once, rather than partially against each other. The nap that feels safe to take at the later hour, because only an hour has passed since the earlier one, is the nap that most reliably deposits the sleeper in the territory where waking becomes difficult. Individual biology shapes all of this. Not everyone carries the same risk from slow-wave awakening, and not all of the variation is circumstantial. Part of the difference between people in how severely and how durably sleep inertia affects them runs through genetics. The adenosine deaminase gene, the gene responsible for producing the enzyme that breaks down adenosine, exists in variants that differ in efficiency. A person carrying a particular variant of this gene clears adenosine from the brain more slowly during sleep. The brake on the homeostatic sleep drive releases more gradually, and the slow-wave sleep that accumulated during a nap dissolves at a different rate. Variants in the genes encoding the adenosine receptors themselves, the A1 and A2A receptor genes, shape how sensitively the brain responds to adenosine binding, and therefore how much homeostatic pressure a given adenosine concentration actually generates. These are not large effects observable with certainty in any individual without measurement, but across populations, they trace a real biological gradient in susceptibility. Age runs along the same gradient. Adolescents and young adults show more severe and more prolonged sleep inertia than older adults, and the reason is grounded in the nature of their sleep architecture. The younger brain generates slow-wave sleep of greater depth and intensity than the older one. The delta waves are higher in amplitude, more synchronized, more fully expressed. The homeostatic capacity for deep sleep is simply greater earlier in life, and the deeper the slow-wave sleep entered during a nap, the more disrupted the awakening from it. An adolescent sleeping in the early afternoon When social schedules, school timetables, and the particular circadian phase shift of adolescence may make this more likely, enters a deeper slow wave episode than an adult in the same conditions, and wakes into a more pronounced and more persistent inertia. These are not failures of the system, they are the system working exactly as it is built to work. Slow wave sleep is deeply recuperative. The brain enters it because it needs it. The inertia is a consequence of being pulled out of a process that was not finished, not a consequence of the process itself being wrong. The question sleep science has spent considerable effort on is not how to avoid slow wave sleep, which would be its own loss, but how to time the nap so that awaking from it is unlikely. And one of the most elegant solutions to this problem arrived not from neuroscience alone, but from the intersection of pharmacology and timing. It begins with caffeine. Caffeine acts as an adenosine receptor antagonist. It does not reduce adenosine in the brain. It occupies the receptor sites that adenosine would otherwise bind. When caffeine is present at the A1 and A2A receptors of the basal forebrain, adenosine cannot produce its sleep-promoting effect there, and the homeostatic pressure it represents goes unfelt. The brain remains alert because the molecule announcing the need for sleep cannot deliver its message. But caffeine taken by mouth does not arrive immediately at those receptors. It must be absorbed through the gastrointestinal tract, enter the bloodstream, cross the blood-brain barrier, and distribute through the central nervous system. This process takes time. The peak concentration of caffeine in the blood following oral ingestion arrives somewhere between 20 and 30 minutes after the drink is consumed. Before that peak, the CNS concentration is still rising. At the moment of ingestion, caffeine has barely begun its journey toward the brain. The caffeine nap exploits this delay. The protocol is precise. A person drinks a cup of coffee or another caffeinated beverage at a dose that approximates the caffeine content of an average cup and immediately lies down to sleep, not 30 minutes later. Immediately, the caffeine, still in the stomach and intestine, still absorbing, still far from the adenosine receptors, offers no interference with sleep onset. The brain, heavy with the genuine adenosine accumulated over hours of wakefulness, slides into sleep. The nap runs for 15 to 20 minutes. And as the nap ends and the person wakes, caffeine is arriving. The timing is not accidental. The nap has partially cleared adenosine from those basal forebrain receptor sites through the natural processes of sleep. Slow wave sleep, with its intense metabolic activity, is particularly effective at this clearance, and even a short sleep produces some reduction. Caffeine arrives at those same sites just as the nap ends and finds receptor occupancy already reduced, which means it can block effectively what remains rather than competing against a full adenosine load. The two effects layer. The nap clears some of the pressure. The caffeine blocks what is left from signaling. The result, measured in psychomotor vigilance testing and in subjective alertness ratings, is greater than either intervention alone. The researchers who studied this most directly were Loughborough University's Jim Horn and Louise Reiner, working in the mid-1990s. Their papers in psychophysiology examined simulated driving performance in sleepy subjects given different interventions during a break. A nap alone helped. Caffeine alone helped. A cold face wash alone helped a little. But subjects who took caffeine and then napped showed the greatest reduction in driving impairment, fewer lane deviations, fewer incidents, better sustained performance compared to any other single condition. The timing was the mechanism. The intervention worked not because caffeine was powerful, but because it was delivered to the adenosine receptor at precisely the moment the nap had prepared the site for it. There is something quiet and precise about this. Two processes, each incomplete on its own, synchronize to complete each other, not stacked, timed. The difference is the 20-minute window between the cup and the waking. The pharmacokinetics of absorption translated, without any calculation on the sleeper's part, into an intervention that is more than the sum of its components. In the dark of a short nap, while the caffeine is still rising in the bloodstream, the adenosine receptors of the basal forebrain are momentarily emptying. The molecular preparation for waking is already underway, and the person asleep on the couch knows none of this. The chemistry is proceeding without supervision, in the space between one waking moment and the next, on a timetable set by biology and by the particular kinetics of a molecule dissolved in a hot drink. The caffeine nap does not eliminate sleep inertia entirely. If the nap exceeds 20 minutes, the risk of entering slow-wave sleep returns, and no caffeine timing will fully compensate for waking from that depth. The protocol depends on keeping the nap short, on waking before the brain descends past the spindles. It is a tool with specific conditions, and those conditions are not always available. Noise, urgency, a body that is more exhausted than expected. Any of these can pull the sleep deeper than intended and carry the nap past the threshold where the caffeine timing still works cleanly. The caffeine nap is therefore not a general solution, so much as a well-understood interaction, a pharmacological fact that can be used when the conditions permit, and that illustrates something about the relationship between adenosine, sleep depth, and the precise window within which a short sleep restores rather than disrupts. But sleep inertia, and the risk of it, is not the only reason some naps fail to deliver what they seem to promise. There is a subtler problem that sits just beneath the surface of the afternoon nap's apparent recovery, one that the sleeper cannot detect from the inside at all. The subjective feeling of restoration after a nap, the sense that the mind has cleared, that thinking is sharper, that the weight of the afternoon has lifted, is real. It is not imagined or self-deceptive in a simple way. Performance on many cognitive tasks does improve after a short sleep, and the improvement is genuine and measurable, but the feeling of restoration and the actual degree of restoration are not always the same thing, and the gap between them can be substantial and persistent. Hans van Dongen and colleagues, working at the University of Pennsylvania, conducted a careful study of what happens to cognitive performance across multiple days of restricted sleep. Subjects accumulated a sleep debt gradually, not through a single night of total deprivation, but through the quiet compression of sleep across successive nights. The kind of reduction that characterizes real life for many people, rather than a laboratory emergency. And across those successive days, cognitive performance declined in a steady, cumulative way. Naps, taken on those restricted sleep days, produced a real and visible benefit. On the performance plots, the curves of declining psychomotor vigilance task scores across the days on which subjects napped, showed performance lifting above the trend line. The napped score was better than the non-napped score on the same trajectory would have been, but it was not better than baseline. It lifted above the declining curve without returning to it. And the next day, without the nap, the decline continued from where it had been, from a point that reflected the accumulated debt, not from a rested starting point. The nap had borrowed against something it could not repay. The deeper problem was in the mismatch between subjective and objective recovery. Subjects who napped reported feeling better. Their ratings of their own alertness and cognitive readiness climbed back toward normal on nap days. But their objective performance, their response times, their lapse rates, their accuracy on sustained attention tasks did not climb as far as their self-assessments suggested. They felt more recovered than they were. And this mattered because a person who feels sharp will act accordingly. They will trust their judgment, accept demands, decline the extra margin of caution that a person who knows they are impaired might choose. The subjective feeling of recovery is itself a function of the brain's current capacity for self-assessment, and that capacity, during periods of accumulated sleep debt, is one of the things most reliably compromised. The nap restored some of the performance. It did not restore all of it, and it did not restore the accurate self-perception of where performance stood. The sleeper emerged from the nap feeling more alert than they were, in a brain that lacked the full resources to notice the discrepancy. This finding does not diminish the value of napping. A performance that lifts above the declining trend is better than one that does not. The short sleep is doing real work, but it clarifies what that work is and is not. A nap does not carry the same restorative weight as the sleep debt it is set against. It buffers the deficit without erasing it. The curve continues to decline. The napped point sits above the slope for a day, and then the slope reasserts itself. What makes this important is not alarm. Sleep debt and its management are facts of life for many people, and a tool that partially helps is genuinely useful. What makes it important is accuracy. The science of napping is most valuable when it is understood precisely. What a short sleep does in the brain, what it does not do, and where the line between restoration and the feeling of restoration falls. The EEG trace of a person waking from slow-wave sleep sits in the record for minutes afterward, the delta waves persisting in a waking brain as visible evidence of a process still running. The fMRI image of the same brain shows the prefrontal cortex lagging behind the brainstem in reactivation, dim in a landscape growing progressively brighter. The behavioral record, the steering wheel's erratic late corrections, the reaction time data with its two slow outliers, translates the neuroscience into consequences that move through the world beyond the laboratory. And the driving simulator trace from a post-SWS awakening, compared to the same subject tested after waking from stage 2, shows the difference as a visible change in a single plotted line. One line flows and anticipates, the other catches up and corrects. The gap between them is the neurophysiology of sleep inertia expressed as a shape on a page, slow wave depth waking as behavioral friction, the brain's incomplete return to itself. The caffeine nap sits at the other end of this picture, not a cure and not a prevention, but a pharmacological timing mechanism that turns the 20-minute window between ingestion and peak CNS bioavailability into a preparation for waking. 2 cups, 1 lie down, 1 alarm set 15 to 20 minutes ahead. Caffeine arriving at the adenosine receptor just as the nap ends, finding the site cleared rather than saturated, blocking what remains of the homeostatic signal before it can rebuild. The sleeper wakes into a brain that has rested briefly and that finds its inhibitory messenger already occupied. The grogginess that might have followed a lighter version of this moment does not arrive, or arrives diminished, or passes quickly. The molecular timing is invisible to the person lying down. It proceeds through the wall of the gastrointestinal tract, into the blood, across the blood-brain barrier, and into the basal forebrain, on a schedule determined by absorption kinetics and nothing else. The sleeper does not manage it. The chemistry manages itself. And the result, measured in reaction times and in the clarity of a mind returning to the afternoon, is a small and precise example of sleep science turned practical, the body's own chemistry used as a tool rather than worked around. There is a steadiness in that, not in the dramatic sense of a discovery overturning something, but in the quieter sense of a mechanism understood well enough to be aligned with. The adenosine receptor does not negotiate. The kinetics of absorption do not vary by intention, but the person who understands the 20-minute window can arrange themselves within it, can drink the coffee and set the alarm and lie down in the knowledge that the chemistry will proceed without further instruction. The biology does not need to be managed. It only needs to be met at the right time. The prefrontal cortex, waking last from slow-wave sleep, dim in the fMRI image, while the brainstem glows with reactivated function. This too is the biology working exactly as it was built to. The ordering of reactivation is not a malfunction. It is the structure of a return, proceeding from the oldest and most essential functions outward toward the newest and most complex, in the same order the brain has always woken up. The grogginess is not a failure of the nap. It is the cost of having been somewhere deep, and the duration of that cost, 5 minutes or 30, depending on the person and the depth and the genetics of adenosine metabolism, is simply the time it takes for the last lights to come back on. The last lights are always the prefrontal ones, the thinking lights, the lights that check the other lights, and for a few minutes, or for a few minutes more than that, the person who has napped too long and too deep sits in a room that feels ordinary while their brain completes a process that has not yet finished. The delta waves run on in the waking record. The steering corrections arrive a fraction of a second late. The subjective feeling of alertness is present and not quite accurate. And then, slowly, the prefrontal cortex comes back. The delta waves thin out of the waking EEG. The corrections arrive on time, and the gap between feeling and function closes. It closes on its own. The biology resolves it, as it always does, on the schedule the biology keeps. The adenosine receptor at the moment caffeine occupies, it is a very small site, smaller than any measurement the body can feel. The molecules that dock there, caffeine on one side of the hour, adenosine on the other, are invisible to every sense the sleeper has. But the consequence of which molecule holds the receptor in the minutes after a nap ends is felt as the quality of the next hour's thinking, as the smoothness of a steering correction, as the accuracy of a self-assessment in a brain that is trying to know where it stands. The science at this scale is a chain of events running from a molecule to a minute, to a measurement, to a decision made by a person returning from a short sleep, to an afternoon that still has hours in it. Each link in that chain is specific and real and follows from the one before. The grogginess is the delta wave persisting. The delta wave persisting is the prefrontal cortex reactivating last. The prefrontal cortex reactivating last is the structure of a return from depth, and the depth was the homeostatic pressure and the circadian phase and the particular genetics of adenosine clearance, all converging at the moment the eyes closed. None of it is random. All of it is physics and chemistry expressing themselves through a brain that has been doing this in some form for as long as there have been brains that need to rest. The caffeine rising in the bloodstream toward its peak is simply chemistry doing what chemistry does, at the rate that absorption allows, arriving at a receptor site that a short sleep has briefly cleared, and blocking what is there with a precision that no one planned, but that the pharmacokinetics made possible. The napper wakes into the quiet aftermath of that event, the mind lifting a little more cleanly than it would have, the afternoon arriving with slightly less friction, the coffee on the table now cooler than when it was poured. The receptor is occupied, the wave has passed. The delta signatures in the waking EEG have thinned and gone quiet, and the brain, all of it, the brainstem and the cortex and the prefrontal regions that check and regulate and decide has returned to the waking world, one layer at a time, arriving last where it always arrives last, settling into the afternoon as the chemistry that shaped the transition completes its work and falls quietly into the background. In the early afternoon, two slow forces arrive together. One has been building since the first waking hour, a chemical accumulation in the brain's deep tissue, molecule by molecule, hour by hour, a rising pressure that has been climbing without pause since morning. The other is older, more architectural, a rhythm embedded in the body's core timing system, a daily trough in the signal that holds wakefulness upright, arriving now as reliably as it has arrived every afternoon for every day of a life. They do not announce themselves. They simply converge, the way two tides moving from different directions arrive at the same shore at the same hour, and the water deepens. This convergence is not random. It is not a personal failing or an afternoon weakness. It is the meeting of a homeostatic force and a circadian, two independent biological processes that happen in the early afternoon to point in the same direction. And the homeostatic force is adenosine, accumulated across hours of waking neural metabolism, pressing against the brain's alertness circuits with increasing chemical weight. The circadian force is the suprachiasmatic nucleus, the brain's master clock, cycling through a predictable dip in its alerting output between roughly 1 and 3 in the afternoon, a dip that belongs to the clock's own inner rhythm and not to any meal, any weather, any effort of will. Between them, they create a window. This window is not a problem to be solved. It is a biological invitation, a time when the brain will accept sleep more readily than almost any other point in the waking day, when sleep onset will come quickly, when the nap will be efficient, when the recovery will be real. The body has been preparing this window since birth. Understanding it does not change it, but it allows a nap taken inside it to be something different from a nap taken anywhere else in the day, something more precise, more restorative, more aligned with what the biology is already trying to do. The adenosine level in the early afternoon is high enough to reduce alertness and accelerate sleep onset, typically to somewhere between 5 and 10 minutes in a person who lies down in a dark, quiet room. This is not the level it will reach by late evening, which is higher still and drives a longer, deeper nocturnal sleep. It is an intermediate level, sufficient to pull the brain rapidly into stage 2 sleep, the light but restorative architecture of a short nap, without yet driving it into the slow, deep oscillations that characterize stage 3. The circadian trough adds to this. The alerting signal from the suprachiasmatic nucleus, which normally pushes back against adenosine's sedating influence and keeps the flip-flop switch biased toward waking, is at its afternoon low. Its resistance is reduced. The balance tips more easily. Sleep arrives. The hours after 3 in the afternoon are different. Adenosine continues to rise, as it always does across the waking day, and the circadian alerting signal begins its late afternoon recovery, the second wind that carries most people through the evening. A nap begun at 5 in the afternoon encounters a different biological landscape than one begun at 1. The homeostatic pressure is higher, which means sleep onset may still be easy, but it also means the brain is more likely to drop into slow-wave sleep sooner and more deeply than it would have 3 hours earlier. And the nocturnal sleep that follows, the long restorative night sleep the body needs, requires its own accumulated adenosine, its own homeostatic pressure, to arrive on schedule and at full depth. A nap taken too late in the afternoon draws from that reserve. It is not that the nap causes harm, it is simply that it borrows from what the night was saving. And in the architecture of the night that follows, the effect is visible. Early, slow wave activity reduced, the deepest sleep arriving later or not at full strength, the long recovery of the night subtly diminished. So the early afternoon window is not arbitrary. It is the point in the day where a short sleep costs the least and returns the most, a convergence that the body's own timing systems create and maintain, and that a nap, taken there, can use completely. The light in the room matters. There are roughly 3,000 cells distributed across the inner surface of the retina that do not contribute to vision in any ordinary sense. They do not form images. They do not detect edges or movement or color in the way that rods and cones do. They are intrinsically photosensitive, meaning they respond to light on their own without needing input from the visual cells around them, and the light they respond to most strongly falls in the blue portion of the spectrum between 450 and 490 nanometers. When blue wavelength light reaches these cells, a pigment inside them called melanopsin absorbs it, and a slow, sustained signal travels from the eye along a dedicated pathway directly to the suprachiasmatic nucleus. This signal does not say, here is a shape, here is a face, here is a moving object. It says, it is day. It says, hold wakefulness. It tells the suprachiasmatic nucleus to suppress melatonin, to elevate core body temperature, to keep cortisol from falling. It is not a visual message. It is a calendrical one, a report about the state of the sky, delivered from the eye to the clock, updating the body's sense of where it is in the day. For a nap to begin quickly and cleanly, this signal needs to be absent, not merely dimmed, but absent. The ambient light in a nap environment below about 1 lux, a level of illuminance roughly comparable to the faint glow of a distant streetlight seen through closed curtains, far below what most indoor environments provide, fails to meaningfully activate these retinal cells. The signal to the suprachiasmatic nucleus quiets. The clock's resistance to sleep onset softens. The transition into stage 1, then stage 2, happens faster and with less effort. This is why even a sleep mask, drawn over the eyes in a bright room, shortens the time it takes to fall asleep. The lids alone do not block enough light to fully silence the melanopsin signal. A proper mask, or a genuinely dark room, brings the retinal input closer to zero. The body reads the absence of blue light as permission. The circadian alerting signal, already lower in the early afternoon trough, drops a little further, and the nap finds its beginning. Temperature is not separate from sleep. It is part of sleep's mechanism. At sleep onset, before a single delta wave has formed, before the first spindle has flickered through the thalamocortical circuit, the body begins to warm its surface and cool its core. The blood vessels in the hands and feet dilate. Warm blood moves outward from the body's interior toward the skin. Heat leaves through the palms, the fingers, the soles of the feet, radiating into the air the way heat leaves a cast iron pan placed on a cool counter, not quickly, not dramatically, but steadily and with clear physical consequence. Core body temperature falls by 0.5 degree to a full degree Celsius, and this drop is not a side effect of sleep. It is part of the signal that initiates it. The ventrolateral preoptic nucleus, the brain region that drives sleep onset, is itself sensitive to this thermal change, and the falling core temperature helps tip the hypothalamic sleep-wake switch towards sleep. The ambient temperature of the nap environment shapes how easily this process unfolds. An environment between 18 and 22 degrees Celsius, roughly 64 to 72 degrees Fahrenheit, allows the body to shed heat through the skin without effort. The peripheral vasodilation proceeds. The core cools. Sleep onset is smooth. In a room warmer than about 24 degrees Celsius, roughly 75 degrees Fahrenheit, the process is impaired. The body cannot shed heat efficiently when the surrounding air is itself warm. The core temperature stays higher than it needs to be, and sleep onset is delayed or fragmented. In a room colder than about 16 degrees Celsius, roughly 60 degrees Fahrenheit, a different problem emerges. The body contracts its peripheral circulation to conserve warmth, redirecting blood back to the core, and the same thermal prerequisite for sleep onset cannot be met. The ideal nap environment is cool enough to allow the body's own thermoregulation to work. The biology asks only that the room be cool and still and dark, that the air have room to carry heat away from the skin, that nothing forces the body to fight against its own preparations. Given those conditions, the thermal change happens quietly, invisibly, in the minutes before sleep, a redistribution of heat moving from inside to outside, the body preparing itself through temperature rather than through any conscious act. Before the body reaches that stillness, the breath can lead it there. 5 minutes of slow, deep breathing, the kind where the inhale expands the lower abdomen rather than the chest, and the exhale is longer than the inhale, and the rhythm is unhurried and low, produces a measurable shift in the autonomic nervous system. The parasympathetic division, which governs the body's rest and repair state, increases its tone. The heart rate slows. Between each breath, there is a slight rise and fall in heart rate, synchronized with the breathing cycle, heart rate climbing fractionally on the inhale, falling on the exhale, a rhythm called respiratory sinus arrhythmia, visible in a cardiac trace as a gentle wave-like oscillation superimposed on the baseline. This oscillation is a marker of parasympathetic health and activity, and its presence, amplified by slow diaphragmatic breathing, signals a shift in the body's internal state away from arousal. Cortisol drops with this shift. The sympathetic activation that keeps the body alert, elevated heart rate, suppressed digestion, heightened sensory sensitivity, gives way to its opposite. The ventrolateral preoptic nucleus, sensitive to this reduction in arousal tone, begins to assert itself more easily. The flip-flop switch, which requires the arousal centers to weaken before it tips into sleep, finds them weaker. Sleep onset latency, the time from lying down to the first unmistakable signature of sleep in the brain, shortens. The nap begins sooner, wastes less of its allotted time on the restless near sleep that precedes it, and spends more of its duration in the stage 2 architecture, where most of the restorative work happens. There is nothing passive about a well-prepared nap. The environment has been shaped. The light has been removed. The room is cool. The breath has been slowed. And each of these preparations has a specific biological target, a retinal cell, a thermoregulatory threshold, an autonomic balance, each one moving one part of the system toward the state that makes sleep possible and efficient. The nap that follows is more productive, not because it is longer, but because it starts cleanly and stays where it needs to be. The question of duration, how long, carries more consequence than almost any other choice in nap practice. A nap of 10 to 20 minutes spends nearly all of its time in stage 2 NREM. The EEG trace during this period shows a characteristic architecture, K-complexes appearing as isolated, sharp waves against a quieter background, sleep spindles waxing and waning in rhythmic bursts, at roughly 12 to 15 cycles per second, the whole record showing the hallmarks of a brain that has crossed from waking into sleep without entering its deepest territories. This is not a superficial sleep. Stage 2 is real sleep. The body's metabolic rate has dropped. The heart rate has slowed. The muscles have released their waking tone, and the spindles that appear within it are not merely markers of sleep depth. They are the signature of a specific neural process, the thalamic reticular nucleus firing in synchronized bursts, sending rhythmic waves through the thalamocortical circuits, each spindle event lasting half a second to 3 seconds, each one part of the mechanism by which the sleeping brain consolidates what it has recently learned and restores the capacity for new learning in the hours ahead. On waking from a 20-minute nap, the subjective experience is typically one of clarity. The adenosine pressure has been reduced, if only partially. The sleep spindles have run their quiet consolidation. The norepinephrine tone that supports sustained attention has recovered. The brain re-enters the afternoon with something measurably restored, not fully, not permanently, but genuinely. Reaction times tighten. Sustained attention holds longer before it lapses. The hippocampus, which had been filling toward its daily encoding limit across a long morning of experience, has been given a small but real clearing, a partial reset of the capacity to form new memories. A 20-minute nap does not replicate a night's sleep. It does not resolve accumulated sleep debt, but within the range of what a short sleep can do, it does almost everything a short sleep is capable of. The nap, that extends beyond about 25 minutes, enters different territory. This is not a failure of the nap. It is simply a matter of biology following its own logic. Homeostatic sleep pressure, still present and still rising even during the nap itself, pushes the sleeping brain toward deeper stages. By the 30-minute mark, in many people under conditions of moderate sleep pressure, the EEG begins to show the first delta waves, large, slow oscillations at less than 4 cycles per second, sweeping in long, high-amplitude arcs through the cortical record. These are the signature of stage 3 NREM, slow-wave sleep, the deepest stage of non-dreaming sleep. The brain at this depth is consolidated, conserved, metabolically slowed. The cortex is cycling through its characteristic alternation of upstates and downstates, hundreds of millions of neurons briefly synchronizing and then falling silent together over and over in slow, enormous waves. Waking from slow-wave sleep is difficult. The prefrontal cortex, the region that governs decision-making, working memory, and complex reasoning, is the last region of the brain to return to full waking activation after stage 3 sleep. Brainstem arousal systems come back first. Primary sensory areas follow. The prefrontal cortex lags, sometimes by 10 or 20 or 30 minutes, its delta wave activity persisting as a measurable intrusion into what should be a waking EEG record. The person sitting up and opening their eyes may appear awake, and in the most basic sense they are, but the most sophisticated operations of their brain are still running in a partial sleep mode. Thinking is slowed, words are harder to find. The sense of time and place returns gradually rather than immediately. This is sleep inertia, and it is not a personal susceptibility. It is a reliable consequence of the architecture that was active at the moment of waking. The nap that avoids slow-wave sleep, kept to 20 minutes or less, avoids this transition entirely. The nap that cannot avoid slow-wave sleep should, if possible, be extended to the other side of it. A full 90-minute nap follows the brain's own ultradian cycle from beginning to end. It passes through stage 1 and stage 2, descends into slow-wave sleep, ascends back through lighter NREM stages, and arrives finally in REM, the stage of sleep defined by near-waking cortical activation, by the suppression of norepinephrine and serotonin, by a surge of acetylcholine that warms the associative and emotional circuits of the brain, and by the rapid, conjugate movements of the eyes beneath closed lids that gave this stage its name. A nap ending in REM does not require emerging from the depths. REM is close to waking. The transition back is smooth, the prefrontal cortex already near its working state, the subjective experience of waking typically clear and sometimes vivid. The 90-minute nap is not practical in every context. It requires time and a suitable environment, and a willingness to commit to a duration that crosses well beyond the boundaries of a quick rest. But when those conditions are available, and first the recovery it provides is qualitatively different from a shorter nap, encompassing the slow wave consolidation, the spindle-mediated memory processes, and the REM-specific operations that belong to emotional integration and the formation of remote creative associations between ideas. It is in effect a compressed replica of one full night's ultradian cycle, offering the brain something close to a full loop through its sleep architecture within a single afternoon hour and a half. The question of what a nap returns to the brain, what specific capacities it restores or enhances, has been studied carefully, and the answers are specific. Matthew Walker and colleagues at the University of California, Berkeley, measured what happens to the hippocampus across a day of learning. In the morning, when subjects studied lists of face name pairs, the hippocampus showed strong spatially clear activation on functional MRI, the encoding process proceeding efficiently, each new pair leaving its trace in the brain's primary memory forming structure. By the afternoon, the same encoding process, with new material, showed measurably reduced hippocampal activation. The brain had not forgotten how to learn, but its primary learning structure had filled toward its daily capacity, its ability to receive and consolidate new information reduced after a long morning of use. Subjects who napped for 90 minutes in the early afternoon showed something different in the evening. Their hippocampal activation during new learning had recovered, not fully to morning levels, but substantially, the capacity for new encoding restored by a sleep that had allowed the spindle and ripple mechanism of NREM to begin the process of transferring recent memories from hippocampal storage toward cortical consolidation, freeing the hippocampus to receive again. Subjects who stayed awake through the afternoon showed no such recovery. Their encoding capacity at the end of the day remained at the reduced level the afternoon had established, the hippocampus still carrying the weight of the morning's learning without the clearing that sleep provides. The performance gap between those who napped and those who did not was roughly 20%, a fifth of the hippocampus's encoding capacity restored by a single afternoon sleep. The mechanism driving this recovery is a nested coupling of three different brain oscillations occurring during NREM. Slow oscillations below 1 cycle per second sweep across the cortex in long organizing waves. The thalamocortical spindles at 12 to 15 cycles per second ride within those slow oscillations, timed preferentially to the oscillation's upstate, its peak of neural excitability. And inside the spindles, originating from the hippocampus itself, sharp wave ripples fire in brief, rapid bursts at 80 to 100 cycles per second, each ripple carrying a compressed replay of a recently encoded experience, a face, a name, a route, an event, transmitted from the hippocampus toward the cortical regions where it will eventually be stored as long-term memory. This three-way coupling, slow oscillation, spindle, ripple, nested inside one another in a precise temporal hierarchy, is not a peripheral curiosity of sleep research. It is the mechanism by which the sleeping brain moves memory from temporary to permanent, and by which the hippocampus clears itself for new learning the next morning or the next afternoon. Attention recovers in a different way, through a different process. The psychomotor vigilance task is a simple measure deceptively so. A subject watches a small screen and presses a button as fast as possible when a number appears. The number appears at random intervals over 10 minutes, offering no warning and no pattern to anticipate. The measure is not how fast the average response is, it is how often the response fails entirely, how many times the number appears and the subject simply does not respond, or responds after more than 500 milliseconds, a delay that in operational terms is the length of a blink extended into something closer to a momentary absence. These are called lapses. They are the cognitive signature of inadequate sustained attention, and they increase predictably and measurably with wakefulness, with sleep debt, with the circadian trough of the early afternoon. A nap of 10 to 20 minutes reduces lapses significantly in the hours that follow. 2 to 3 hours of improved sustained attention following a sleep that lasted less time than a standard lunch break. The neurophysiological basis for this recovery involves the locus cerebellosus, a small nucleus in the brainstem that is the brain's primary source of norepinephrine. Sustained wakefulness depletes the tonic norepinephrine release on which sustained attention depends. Even brief stage 2 sleep partially restores this tone, and with it, the ability to maintain the kind of alert, focused waiting that the PVT demands. The adenosine that had been accumulating in the basal forebrain, inhibiting the cholinergic neurons that support cortical arousal and attention, is also partially cleared during the short nap. The combination of reduced adenosine inhibition and restored norepinephrine tone is enough, following a short sleep, to produce a measurable and meaningful improvement in how long a person can sustain attention before it begins to fail. Motor learning recovers through a mechanism that is visible, specifically and spatially, in brain imaging during sleep itself. In studies of finger tapping sequences, where subjects practice typing a specific series of keystrokes rapidly and accurately, repeating the sequence hundreds of times before resting, the performance gains measured after sleep are not simply the result of passive consolidation. During non-REM sleep following the training session, the motor cortex, the primary cortical region responsible for executing the learned sequence, shows task-related activation patterns that mirror those recorded during the waking practice. The sequence is being rehearsed. Not consciously, the sleeper has no awareness of it. But in the neural record, the spatial pattern of activation during sleep matches the pattern of practice, and the magnitude of this offline rehearsal predicts how much performance will have improved when the subject is tested after waking. The brain is using sleep to practice what the body learned while awake. When this practice occurred within an afternoon nap rather than an overnight sleep, the performance gains were comparable, roughly equivalent to a full night's benefit in a 90-minute window. The nap was not a pale substitute for full overnight sleep. In terms of motor sequence consolidation, it accomplished what the night accomplishes, given comparable architecture. The stakes of attention and motor performance are not always confined to laboratory measures. In the 1990s, researchers working with NASA's Ames Research Center studied commercial airline pilots on long transoceanic flights, the kind of flight where the cruise phase stretches for many hours at altitude. The autopilot manages most of the flight path, and the most critical work arrives at the end, during approach and landing. Pilots were monitored with continuous EEG throughout the flight. In the cockpit record, among pilots who were not given a scheduled rest opportunity, a particular signal appeared with regularity, intrusions of delta and theta activity into what should have been a waking, alert EEG trace. These intrusions lasted seconds. They were distinct in the record, a sudden disruption of the normal desynchronized high-frequency pattern, replaced briefly by the slow, large amplitude oscillations of sleep. The pilot's eyes were open. They were seated at the controls, but their brains were, by every neural measure, momentarily asleep. These episodes are microsleeps. They are not the result of any unusual fatigue or individual vulnerability. They are the predictable consequence of sustained wakefulness in a warm, vibration-dampened, low-stimulation environment during the circadian afternoon trough. The brain, under these conditions, takes what sleep it can find in fragments without permission. Pilots who were given a planned rest opportunity during the cruise phase, a 40-minute window in which they could recline with a sleep mask and attempt to nap, slept for an average of 26 minutes. The rest did not resolve their accumulated fatigue. It did not replicate a full night's recovery. But when their performance was measured during approach and landing, the phase that demands the highest precision and the fastest response, those who had napped showed a 34% reduction in microsleeps compared to those who had not. Their reaction times improved by 16%. The difference between a brain that had been given 26 minutes of stage 2 sleep and a brain that had not was measurable in the record, visible in the EEG, and expressed in the precision with which a pilot brought a wide-body aircraft toward a runway. The recovery a nap provides is real. It is also bounded. Hans van Dongen and colleagues at the University of Pennsylvania conducted a series of studies in which healthy adults were restricted to 4 or 6 hours of sleep per night across multiple consecutive days. The cognitive impairment that accumulated across those days was progressive and consistent, each night of reduced sleep adding to a growing deficit in sustained attention, working memory, and processing speed that the subjects themselves increasingly underestimated. By the 4th and 5th days of restriction, their performance on the psychomotor vigilance task had declined to levels equivalent to complete sleep deprivation. Their subjective sense of sleepiness, reported each morning on standardized scales, had largely stabilized. They felt roughly as sleepy on day 5 as they had on day 2, even as their objective performance continued to deteriorate. Naps inserted into some days of the restriction schedule produced a temporary improvement. In the performance data, plotted across successive days as a declining curve, the nap days appeared as points lifted briefly above the general slope, a recovery visible as a small upward deviation from the underlying trend. The nap was doing something real. The lapse rate was lower. The reaction times were faster. By that single day's measure, the nap had helped. But the following day, without another nap, the performance fell again, not to where it had been on the day before the nap, but to a level that continued the underlying declining trajectory, as if the nap had granted a single day's reprieve without altering the slope of what was happening beneath. The accumulated sleep debt was not resolved. The nap had not repaid what the nights had failed to provide. It had delayed the expression of the deficit, smoothed one point on a deteriorating curve, and then receded as the biology resumed its course. There is something important in this. The subjective experience after a nap on a sleep-restricted day is one of genuine recovery. The person feels better, more alert, more capable, and by the objective measure of that afternoon, they are better. The nap's benefits are real at the moment they occur, but the feeling of recovery that follows a nap can exceed the actual recovery. The gap between how a person feels after a short sleep and how they actually perform on demanding tasks, particularly on the sustained attention measures that are most sensitive to cumulative sleep debt, is not small. A nap taken during a week of poor sleep produces a person who feels more alert and actually performs somewhat better, but who still carries the underlying deficit forward into the days ahead. The deficit invisible to them because their subjective sense of sleepiness has stopped tracking the objective reality. What a nap is, settled cleanly, is this: a partial and temporary recovery within the architecture of a single day. Its benefits are specific: attention, hippocampal encoding capacity, motor consolidation, the reduction of micro-sleep episodes, and measurably real within the hours that follow. Its limits are equally specific. It does not resolve accumulated sleep debt. It does not substitute for the long recovery of nocturnal sleep, and the confidence it restores can outpace the performance it actually repairs. Both the benefits and the limits are expressions of the same underlying biology, the same adenosine chemistry and thalamocortical architecture and homeostatic accounting that govern every hour of sleep in every form. Understanding both is not a counsel of discouragement. The nap's benefits are genuine. The afternoon window is real. The 20-minute limit and the early timing and the cool dark room and the 5 minutes of slow breathing before lying down, each of these has a specific target in the biology, a mechanism it is adjusting, a process it is enabling. The optimal nap is not a myth of self-improvement culture. It is the result of taking the body's own system seriously, of aligning a deliberate behavior with processes that have been running in the human brain since long before anyone thought to measure them. The performance curve from Van Dongen's studies shows something else besides its decline. It shows the nap point rising above the trend, clearly, visibly, a single day where the biology was given what it needed and responded accordingly. The curve then falls again. The arc continues. But that lifted point remains, a record of what the body can do when a short sleep is timed well, prepared for, and allowed to complete its work. The early afternoon light falls across a quiet room. The curtains are drawn, and the temperature is cool, barely perceptible against the skin, and the air is still,